Morning, good afternoon, and good evening, everyone. Welcome to session 3 of the workshop, the last and the best session. People say the best comes last, right? Which is the update on FDA's guidance and recommendations for assessing formulation stability and approaches to bioequivalence. My name is Bing Li. I'm the Associate Director for Science in Office Bioequivalence, Office Generic Drugs. And joined me are Dr. Khondoker. He's a senior pharmacologist in Office Research Standards, who is also the key organizer for this nitrosamine-related workshop. And also Dr. Zhang, Associate Director for in Division of Bioequivalence 3 in Office Bioequivalence, and the 3 of us together will co-moderate this session. So in our previous session yesterday and this morning, by the way, they are also best sessions. We heard speakers discuss important safety and risk considerations for acceptable intake limits, the factors contributing to the formation of NDSIs, the advancement in analytical methods to assess nitrosamine impurities, and various strategies to mitigate these risks. And throughout these talks, what we can see is that over the past 6 years, the FDA and industry have dedicated a significant effort to addressing the risk posed by nitrosamine impurity in drug products. We see more and more clear picture on setting up acceptable limit, quantifying the nitrosamine impurities, and strategies in mitigating these risks. With these efforts, pharmaceutical industry are better positioned to redevelop their product to produce a more safe and clean product with acceptable nitrosamine impurity levels. Now, the question is, after making changes and redevelop these drug products to ensure the safe nitrosamine level, how can we demonstrate that they are still equivalent to their original product? Additionally, with a substantial number of new drug and generic drug applications affected by nitrosamine issue, is there a streamlined approach to demonstrate the bioequivalence of these modified products? And if so, how can this approach be implemented efficiently in the application assessment process? And moreover, what challenges are remaining and what future work lies ahead? These questions form the focus of our discussion in today's session 3. I invite you to keep this point in mind as we proceed. So in the next few hours, you will hear speakers from FDA, industry, and academic who will share their insights and provide updates on a recently published FDA guidance, the second version of guidance for industry, control of nitrosamine impurities in human drugs, released on September 2024, just 2 months ago. And our speaker will navigate you through the most advanced FDA's recommendation for controlling nitrosamine impurities, as well as introducing a streamlined BE approach when reformulation takes place, outlined in the guidance. And following that, our speaker will discuss the practical considerations for implementing these strategies in NDA and ANDA submissions with highlighted case studies on various scenarios. Additionally, you will learn about recent findings on the impact of antioxidant on drug permeability and reformulation considerations. Finally, the speakers will share some exploratory work on bioequivalence risk assessment and PBPK modeling to support bioequivalence consideration for BCS4 and modified release product, which are the challenged product of the category. And at the end of the session, we'll invite you to join a panel discussion where our panelists will address questions from the audience and provide their insights on topics covered in today's session. I encourage you to ask questions to foster an interactive discussion. Now, without further ado, I'd like to invite Dr. Khondoker to podium to introduce the speakers and their presentations. Alam, please.
Thank you, Bing. We have many speakers from FDA across different office, different division. We have speakers from academia and also from industry. So it will be a really nice session. I will briefly describe their background so that you can ask a relevant question. And if you are interested to learn about their full background, please go into the CRCG website. You can find them there. So our first speaker will be Susan Zhang. Ms. Susan Zhang currently serves as a supervisor in the Division of Regulations and Guidance. In this role, Susan oversees the development of policies in a number of quality-related areas. Dongmei Liu, Dr. Dongmei Liu is currently serving as Acting Deputy Director in the Division of Therapeutic Performance 2, that is also known as DTP2. She was primary author of the FDA guidance for industry titled Control of Nitrosamine Impurities in Human Drugs that was just published in September 2024. And Dr. Liu also led a couple of FDA-sponsored research projects on nitrosamine mitigations. Our next speaker will be Dr. Jim Polli. Dr. Polli is a professor in industrial pharmacy and pharmaceutics at University of Maryland. His research interest is oral drug absorption involving laboratory and clinical research. He has served as advisor to 25 PhD graduates. He is also serving as co-director of the University of Maryland Center of Excellence in Regulatory Science and Innovation, that is also known as SARC, and also co-director of CRCG. Our next speaker will be Dr. Wang. Dr. Wang is Associate Director in the Division of Bioequivalence 1 within Office of Bioequivalence. Dr. Wang has actively engaged in cross-office and intra-office working groups focused on nitrosamine-related issues. Our next speaker will be Dr. Hansong Chen. He is currently working as Senior Interdisciplinary Scientist at the Division of Product Quality Assessment 12 within OPQA 2. He has vast experience on different regulatory aspects by working in different offices within FDA. Our industry speaker, Mr. Sivacharan Kollipara, he is currently working as biopharmaceutics team lead in the Global Clinical Management Group, IPDO, at Dr. Reddy's Laboratory Limited, Hyderabad, India. He is responsible for biopharmaceutics evaluation, bioequivalence risk assessment. He is also involved in PK modeling simulation activities to support the generic drug development program in his company. Our next speaker will be Dr. Zhang. Dr. Zhang is serving as team lead for the immediate release drug product team in the Division of Therapeutic Performance 2 within Office of Research and Standard. In her current role, Dr. Zhang works with a group of interdisciplinary scientists developing product-specific guidances and communicating with industry through various mechanisms. Our next speaker will be Dr. Fang Wu. She is currently serving as senior pharmacologist and scientific lead for oral PPK team in the Division of Quantitative Methods and Modeling, also known as DQMM, within Office of Research and Standard. She is responsible for using modeling and simulation tool to address regulatory questions. We'll have additional members in our panelist, Dr. Liang Zhao, Dr. Bhagwant, Rege, and Dr. Sumit Madan. I will introduce them before the panel discussion. Now, I want to welcome Mrs. Susan Zhuk to present her work.
Let me make sure I know how to use this. Okay. Good afternoon, and I want to thank Alam for inviting me to speak, and Dongmei as well, and thank you for the kind introduction, Bing. I'm Susan Zhuk. I'm from the Policy Office in the Office of Pharmaceutical Quality, and I'm going to share this presentation with Dongmei Liu. She's from the Office of Generic Drugs from the Office of Research and Standards. And even though we're from two different offices now, for the past 6 years, we've worked on nitrosamine projects together, especially with policy development, including working on the guidance for industry. So in this session, we're going to be focused on the 2024 Control of Nitrosamine guidance, and we're going to highlight some of the key new information that came in that guidance. Got an image of the guidance on the side in case you'd never seen the guidance before. I'm going to go over a little bit of the history of nitrosamine guidances at FDA, the new recommendations that are found in the 2024 guidance, how industry can best implement the recommendations that are made in that guidance. And Dongmei is going to be talking about alternative approaches to bioequivalence when a manufacturer decides to reformulate their product to mitigate nitrosamine impurities. And then we'll end with some takeaway messages. So the first nitrosamine guidance was published in 2020, and there was a minor update in 2021, just to add a timeline, actually. In that guidance, if you think back to 2020, what we knew at that time about the root causes of nitrosamine impurities was really focused on small molecule nitrosamine impurities, such as NDMA. That's what we knew about. And what we knew about the sources really was focused on the synthesis of the API and possible nitrosating events that would happen during that synthesis or contamination from solvents and such. In that guidance, we first introduced the three-step mitigation strategy that has continued to be present in subsequent guidances. And that mitigation strategy starts with you conduct a risk assessment to see if there's a risk of a particular nitrosamine forming. You perform confirmatory testing to see if that nitrosamine does form and at what levels. And then step 3 is to report to the agency when changes need to be made to mitigate the nitrosamine impurity. And in that original guidance, as I said, we only were focused on small molecule nitrosamines. In fact, we had a little short list of known small molecule nitrosamines. In 2023, the rail guidance was published, and there's been a lot of discussion at presentations about that guidance. That was a different kind of guidance. It was focused on pharmacology and toxicology. It provided a new framework for determining the acceptable intake of nitrosamine impurities, and it was focused on NDSRI type nitrosamine impurities, and it gave us the CPCA approach. In 2024, we published in September this new version of the control of nitrosamine guidance. This is a CMC guidance. It's a quality guidance. We had learned a lot in the 4 years since the original publication of this guidance, and so we had broadened the scope of the guidance. We included both small molecules as well as NDSRIs, describing root causes of both of those formations. We added a lot of new recommendations, and one thing I want to say about those new recommendations is that what we recommended was largely due to input that we got from industry after the publication of the first guidance. You know, we learned where industry was having difficulty in applying our original recommendations, and so we made changes. That guidance also provides a whole new section on implementation of recommendations. Again, this was to help industry understand what FDA expects. And lastly, it included a large section on reformulation through use of antioxidants and pH adjusters, and it gave some new suggestions for bioequivalent studies. I'm going to talk in the next 3 slides about some of the new recommendations in that guidance that we think are useful to industry. The first of those was described in great detail by Dan Berger yesterday, and that's how to set a total nitrosamine impurity limit when you have multiple nitrosamines present. Now, the 2020 guidance gave a very strict limit if multiple nitrosamines were present. It said that if you have multiple nitrosamines, the total limit should be based on the most potent of those nitrosamines. And that often resulted in the total nitrosamine limit being 26.5 nanograms per day. We heard from industry that's very hard to meet. In the new guidance, we've introduced a flexible approach to total nitrosamines. Dan went into a great deal of detail on this and gave some good examples, so I'd like you all to refer to his presentation for that. But suffice to say that instead of a very strict limit based on the most potent nitrosamine, a manufacturer can look at the individual AI limits for the nitrosamines and look to see whether or not the nitrosamines based on the percentage add up to more than 100%. As long as the percentages add up to within 100%, that is a passing result. Another change that we made based on feedback from industry was in our recommendation for sensitivity of analytical methods used to detect nitrosamines. We had, regarding the limit of quantitation, a recommendation in 2020 that the LOQ should be 0.03 parts per million, no matter what the AI is. That was very difficult for a lot of manufacturers to meet. In 2024, we introduced a more flexible approach. We recommended that manufacturers use the recommendations of ICH Q2R1, which says that both the LOQ and the LOD should be commensurate with the target for the impurities that are being detected. So the target for a nitrosamine impurity would be the AI. That's your target limit. And I've given a little example of how this differs on the left-hand side of the slide. So for a drug, a made-up drug, that has MDD of 100 milligrams per day and an AI of 1,500 nanograms per day, that the control limit for such a drug would be 15 ppm, and that's just the conversion of the 1,500 nanograms to ppm. So per the old guidance, the LOQ would have, the recommendation would have been 0.03, even though the AI is quite high. In the recommendations in the 2024 guidance, if a manufacturer, for example, chose an LOQ that was 10% of the AI, which is a reasonable way to set an LOQ, the LOQ could be 1.5 ppm. So you can see that's a difference that would be helpful to manufacturers. It gives a lot more flexibility for the method sensitivity. I'm going to talk a little bit about that 3-step mitigation strategy here, and I hope you can follow along with me. So just as a reminder, the first step in mitigation is to identify the risk, and in my little flow diagram, that's the blue boxes. And the next step is to perform confirmatory testing for that nitrosamine that's found to be at risk, and that's the orange boxes. And then the last step would be to report to the agency if changes need to be made, which is the kind of manila shaded boxes at the bottom. So starting at the top, the guidance, and there's a whole section on implementation in the new guidance. So the guidance says that the manufacturer identifies the risk for a particular nitrosamine. Now, in the old guidance, we talked a lot about the manufacturer identifying the risk. But now we have a web page where the FDA is also identifying risks. So the risk that's identified could either be from the manufacturer or from the FDA. The first thing that the manufacturer should do is to determine whether this risk really exists. And I know Andre alluded to this morning, but the new guidance says that the manufacturer can perform stress testing of the API, and Dr. Sharma talked about a kind of testing that he is working on. So the manufacturer can perform stress testing and prove to us this nitrosamine doesn't form. If that is the case, then no further work needs to be done. The manufacturer does not need to set a specification limit for that particular impurity, even though it was identified initially as a risk. The manufacturer can prove that indeed it does not form. In the absence of that data-driven study, the manufacturer should go on to the next step, which would be to perform confirmatory testing. And now I'm going to go on to the orange boxes. So once confirmatory testing is done, there's 3 possibilities for test results. I'm going to start at the left here. So the first possibility is that the test results are very low, that they're less than 10% of the acceptable intake limit. If that is the case, then the manufacturer need not establish a specification, need not perform routine testing for that impurity. The only caveat here is that if manufacturing changes are made, the manufacturer should revisit this and reperform the testing. The second possibility is that the nitrosamine forms above 10% of the AI, but it's within the AI limit. In such a case, the manufacturer should establish a specification for the product and should perform routine testing for that impurity. And the last possibility is that the test results show that it's above the AI. In that case, the manufacturer is likely to need to make manufacturing changes. Those changes should generally be submitted to the agency through a prior approval supplement. The other alternative for manufacturers is you can always propose a new AI that's going to have data behind it to support the new AI. And the guidance does go into a lot of detail about what kind of studies have to be performed. That kind of supporting data for a new AI should come to us through a prior approval supplement. I'm not going to go into much of the timeline stuff, but so you should pay attention to the date at the top of the slide, August 1st, 2025. Both the rail guidance and the new nitrosamine guidance inform industry that by August 1st, 2025, the testing for NDSRIs, as well as any reporting of changes made in the manufacturing process, should be submitted to the agency. And the last thing I'm going to leave you with is a screenshot of the website that's associated with the guidance. The web page itself is actually considered a guidance of sorts. And the website is the same website as was used for the rail guidance. We now share it between the 2 guidances. This website is a big benefit. It allows us to readily make updates on AIs, on information for industry about nitrosamines as we learn about it. And I want to bring your attention to, I think it's the fifth bullet, emerging scientific and technical issues. Somebody this morning mentioned an announcement about ritonavir. It's the first time that we've used this new section of the website in order to announce that emerging issue, and we expect to use it more in the future. And the last thing I want to leave you with is that little gray sort of toolbar in the middle of the bottom of the slide. That's how you can subscribe to the website so that you can get updates as they occur. I would encourage everybody to put in your web address or your email and get those updates, because we have made several updates even since September when we first posted the guidance, and we intend to make more. So this way you can know about new AIs, interim limits if we publish them, and other really important information about nitrosamines. And now I'm going to turn it over to Dongmei Lu to talk about mitigation through reformulation. Take it away.
Oh, thank you, Sue, for the information about the new recommendations in the FDA nitrosamine guidance. So next, I'm going to give the information about the NDSI and mitigation strategies and FDA's recommendation on the reformulated drug product. So FDA's recommendation for the NDSRI mitigation strategy including the following. The first one is to screen the excipients using the supplier's qualification program for the potential nitrite impurities across different excipient suppliers and excipient lots. In terms of the reformulation, FDA's recommendation to the applicants is to add antioxidants or add a pH modifier in the drug products. Because several research already indicate that in a very small amounts of antioxidants actually can significantly inhibit the nitrosamine formation, including NDSRI. For the pH modifier, you already know, and because it can modify the microenvironment in the drug product to neutral or a little bit basic pH in the drug products. So in this way, the nitrosamine formation, the kinetic reaction is slowed down, slowed down a lot. Also, FDA recommends, also encourage industry to have an innovative approach. And when we talk about this reformulation by adding antioxidants or pH modifier, in general, that is considered as a major change. And stability data and bioequivalence data need to be submitted to support these changes. In terms of the stability data, amounts of the stability data, that really varies. That really depends whether the nitrosamine level has been demonstrated to be stable, especially at the early time points. And due to the lack of the historical information about NDSRI, FDA recommends 3 drug product batches should be submitted to support these changes. And at the time of the supplement submission, 3 months of the data, including the accelerated stability with the time points of 0, 1, 2, 3 months, and the long-term stability data with the time points of 0, 3 months should be submitted at that time. FDA may request 6 months accelerated stability data before approval if the analysis is showing that for the nitrosamine impurity, there's a potential it may have this upward trend, which may lead to out of a specification results. In terms of the long-term stability data, for the first 3 batches, and this data should be reported through the proposed shelf life in the annual report after the approval. And also, when we talk about the OTC monograph drug products, manufacturers should have the similar approach and should retain the similar documentation at their facility site. FDA recommended alternative bioequivalent approaches to the reformulated drug products. According to the current SUPAC guidance, if add this antioxidant or pH modifier into the drug products, belongs to level 3 changes. So that is the challenge, because for level 3 changes, an in vivo B study generally is needed to support this change. But in terms of the regulations, it provides the type of acceptable evidence and available approaches to establish BA and BE, including the regulation listed right here, defined in the 21 CFR 320.24b6, any other approach deemed adequate by FDA. In terms of the scientific evidence, FDA either conducted or sponsored research project and to give this information, the scientific evidence in this perspective. And the first project I want to talk about, that is FDA's internal project. The result indicate that addition of antioxidant to the formulations could significantly inhibit nitrosamine formation. And the second and third, that is related to the bioequivalence. The second one is the research project indicate that the small amounts of 4 antioxidants, including ascorbic acid, alpha-tocopherol, propyl gallate, or cysteine hydrochloride, they don't significantly affect the permeability of the BCS3 model drugs. And the third project indicate that the small amounts of antioxidant do not affect the intestinal transporter activities, and about 30 antioxidants were studied in the third project. And because you notice that in this permeability testing, actually we use this BCS3 model drugs. It has this feature of this low permeability. Actually, that is the worst case of the scenario to study the excipient effect. But for BCS1 and 2 drugs, and which have a high permeability, so the antioxidants would be expected to have no effect or minimum effect on the permeability of BCS1 and 2. So that's the reason why the results of here, I indicated right here, that could be for BCS3 drugs, that could be extrapolated to BCS1 and 2 drugs. And also, I want to indicate that in Dr. Polli's research project, and he's going to give the presentation in the following presentation, and also provided information, similar information that the antioxidant do not affect permeability of BCS3 model drugs. And so FDA established the policy on this alternative B approach, and it separates that into 3 categories. The first category, if the immediate release solid oral drug products or oral suspension drug products with the API as is the BCS1, 2, and 3, and the reformulation including a pH modifier or one of the specific antioxidants studied in the FDA's research, and the antioxidant amount is no more than 10 milligram per dose or maximum daily exposure, whichever is lower. So for this category 1 conditions, FDA's recommendation is comparative dissolution testing data in the QC, in the quality control medium and multi-pH 1.2, 4.5, and 6.8 on the pre and post change products. In general, that should sufficiently support this change. Category number 2, also immediate release solid oral drug products and oral suspension. This is a reformulation and it incorporates one of the specific antioxidants which studied in the FDA's research, and the antioxidants in amounts of more than 10 milligram per dose or maximum daily exposure, whichever is lower, or if the added antioxidants it is other than the 4 antioxidants studied in the FDA research. So the recommendation for this category 2 also separated into 2 scenarios. The first one is for BCS1 and 2 high permeability drug products. In general, dissolution data in the QC and multi-pH should be sufficiently to support these changes. And in very limited cases, additional study such as in vitro bridging study or in vivo B study might be requested. The second scenario, it is for BCS3 low permeability drugs. And for this scenario, supportive studies to demonstrate that antioxidant doesn't affect the passive intestinal permeability and the transporter activity. If the API, it is the transporter substrate. And dissolution data in the QC and the multi-pH should be submitted to support these changes. If this information is not adequate to support the changes, more supportive information such as in vivo BE study would be recommended. So category number 3, for this IR solid oral or oral suspension drug products, so which is the API as is the BCS4 classification, or modified release drug products. FDA's recommendation are as follows. So in general, the alternative B approach for these conditions are not applicable. And, but the applicants, you can submit IVC or PPPK information to support your change. Or, the worst case of a scenario, you can still submit the in vivo BE study to support this reformulated drug product. And FDA encourages applicants to communicate with the agency on the novel BA and B approaches. So for this revised guidance, as a conclusion, I want to give you this take-home message is that this guidance describes 2 structure classes of nitrosamines. You all already know that it's a small molecule nitrosamine and NDSRs. It also provides potential root causes of NDSRs and along with its control and mitigation strategies. This guidance added new recommendations for nitrosamine impurity controls. It also clarifies how manufacturers should assess the test result to determine whether the specification is needed for your case and its reporting channel to the agency. Alternative approaches to establish total nitrosamine impurity limits is included. The guidance also provided implementation recommendations, including to include this NDSRI in the 3-step mitigation strategies. Along with the recommended timeline. And it's also included reformulation as a mitigation strategy, which is the CMC stability information and the bioequivalence recommendations. And then finally, it's provide a web page for the nitrosamine acceptable intake limits. And so finally here, I want to acknowledge our colleagues in OPQ, in OND, and OGD. And my colleagues really help us a lot with this research project and also with this guidance development. And also the colleagues in multiple nitrosamine working groups in FDA. Thank you so much for your attention.
Excellent presentation by Ms. Susan Zook and Dr. Dongmei Lu. So now I like to welcome Dr. James Polli.
Thanks, Alan. Okay, what I'd like to speak with you about is use of antioxidants for reformulation and implication of added antioxidant on drug permeability. So it's largely, there's 3 bullet points I kind of want to cover. I'm mostly spending time on the second one. But very briefly, first we'd like to talk about context of biopharmaceutical risk in potential CMC changes. Talk about the studies that we've done, which is the first reference there. Yuli's in the back, and she did this work. And then also listed there are the 2 references that are in the guidance that I think you're probably already familiar with. And also, Root 2 is a graduate student in the back, and she's recently done some comparative dissolution evaluation. And in my mind, there is a connection between at least the first bullet and the 3rd bullet in terms of comparing dissolution profiles. It might depend upon what the change is. And so what are some guidances that are out there that deal with biopharmaceutics and CMC changes? They're listed here. There's M9BCS, there's SUPAC-IR, that has elements of biopharmaceutics in it, as well as, of course, the recently updated control of nitrosamine impurities one. And one thing about the recent one, which is really a great advance, I think we could all agree, is that it concerns some very limited, compared to other guidances, some very limited CMC changes, right? In fact, some of you probably complain that it's not explicit about enough additional changes. But if you compare the nitrosamine impurity guidance from a CMC and biopharmaceutical risk standpoint, say to BCS, where there's, under some circumstances, extremely broad potential for CMC changes, right? Like a class 1 drug. So when we think about this guidance, I'm not sure we should always read in everything that we know about other guidances, because clearly this guidance is much more limited, I think, in terms of biopharmaceutical risk. Okay, so let's mostly talk about some drug permeability studies that Yuli did. So here's kind of a summary. Okay, so whoops, can't get the mouse to work. Sorry. Okay, so we did some studies using MDCK monolayers, which are a highly established permeability model, very similar to KCO2. In fact, they often give very similar values. Just to show you what these look like, so here's the cells grown on a transwell format. That's a certain type of plasticware that you can purchase. In this case, it happens to be 6 wells. You can also buy it in 24 wells, et cetera. And because of the way the transwells are arranged, there's an upper chamber as well as a lower chamber. And the cells are grown on the, this pointer's not working too well. The cells are grown on the bottom of that insert that's sort of prominently carried there. And the cells are very smart. Biology is amazing. There's an inside and an outside. There's a apical side outside the body, say drug lumen, and then there's a blood side, the basolateral side. And we did studies for drugs in the presence of absence of 10 different antioxidants in both the apical to basolateral, as well as the basolateral to apical. We did this for 4 drugs, ranitidine, cimetidine, acyclovir, and pyrazenzapine. We studied 10 antioxidants, as well as a positive control SLS, sodium lauryl sulfate, a detergent, which is well known to disrupt monolayers in vitro. And then we calculated a permeability value, which is shown there. And other metrics that we kind of looked at were ratios. We looked at the efflux ratio in terms of the basolateral to apical permeability versus the apical to basolateral, since we did it in both directions, and also the relative permeability in terms of the, and all this is published in that article at the bottom, in the presence of excipient as well as the absence of excipient. Okay, and then in terms of, it's actually very hard to assess excipient effects, I would say. It's very attractive to maybe use a t-test, but I think you'll probably get a lot of positive controls. Lots of labs historically are very familiar with this type of testing. So we use a totality of evidence approach. So a couple different things. Drug permeability values with and without antioxidants were obtained on the same day. And in terms of results, in terms of questions that we ask, did the antioxidant modulate drug permeability twofold for each apical to basolateral or basolateral to apical? And the answer was no. And there's a reference at the bottom to an FDA guidance on transporters and metabolism. That's a basis for that twofold value that I can answer questions about. And then another question we ask is for each antioxidant, was there a trend? Right, we're studying, for each antioxidant, we're studying various drugs. Was there a trend in antioxidant to modify permeability beyond a relatively small range, 80 to 125%, across the 4 drugs? And the answer was no. Of the potential 80 antioxidant drug pairings, if you think about 10 antioxidants, 4 drugs, 2 directions, there really was no trend at all. In terms of this 80 to 125 ratio. In fact, there was only one pair, para-aminobenzoic acid, 4-aminobenzoic acid, with one of the drugs and it only happened twice. So we would not call that a particular trend. So here are just some some of the most outstanding values that we saw, which were not outstanding at all, actually. So the relative permeability was typically between 80 and 120% with no pattern. Our positive control, of course, blew that away, very much so. Not that SLS causes these effects in vivo. In fact, we've shown that it hasn't, but it's still a positive control for the purposes of this study. The largest apical to basolateral permeability that we saw relative in the absence of surfactant was 1.44 for one particular drug excipient pair. The smallest was a different excipient pair. The largest relative basolateral to apical was yet a different drug excipient pair. And the smallest was actually the same as the smallest in the other direction. So in general, we say there is just simply no evidence of really an excipient effect. And just a little bit more about our positive control. Meanwhile, our positive control, as you'll see, was always much higher than any of these values that you see here, if you're not familiar with these permeability ratio things. And just so even though SLS we're using as a positive control, we've done PK studies where we've demonstrated, I forget how much of it's 25 milligrams or even more of SLS has no effect in vivo, even though it has a very large effect in these in vitro studies, which is good. So these in vitro studies are sort of like a canary in the coal mine type of approach. Again, we're using MDCK cells, structures there for the four. These are very familiar BCS class 3 prototype drugs. And the antioxidants, the 10 antioxidants are listed on the right side. I think this is with maybe 10 milligrams per tablet would be the way in which we constructed these concentrations. We used some chromatography and liquid scintillation counting. And so the next couple of slides are just kind of results. They all kind of look the same because there was just really nothing remarkable. Just looking at the maybe a little bit easier to read the right-hand side. This is ranitidine apical to basolateral, so sort of absorptive permeability from the GI tract into and across the cells. That's the apical to basolateral direction, and the graph below it is the opposite, the basolateral to apical direction. And the very first one there on the left-hand side is the control, i.e. without excipient. These are permeability values. And then on the extreme right is the positive control, sodium lauryl sulfate. Not that sodium lauryl sulfate at these concentrations causes any in vivo effects. And there's really just not too much difference. And that was repeated. That's basically seen the same with regard to basolateral to apical. And then this is the same type of situation for cimetidine. There's a couple things here in red, I think because they're outside the 80 to 120 range or something like that. So most notably, there was, again, we kind of look at the outliers to the extent that they're outliers the most, and again, we don't see any trend. And then the same thing with acyclovir, although I think there was actually nothing here to really look at with any note. And then the fourth drug here. There was a general trend in terms of the controls tended to be a little bit higher than all of the tests for both directions, which implicates that it was really just an experimental thing more than anything, which happens quite a bit with these type of studies, which is why we avoid t-testing, well known to avoid t-testing. Okay, and then here's just kind of a summary, and again, you can look at this, it's in the paper, and I'm not sure you'll really see much of any trend. So we concluded there was really no effects. Now this is really more passive permeability. So in the guidance, it does talk about, considering transporters. None of these drugs are notable for having any major in vivo transporter effects. Not that you can't necessarily measure them in cell culture. And just based on the totality of looking at the data and considering the excipients, one situation that we at least thought about, try to give some consideration to, was the drug pirenzepine in the context of the antioxidant cysteine. Now, cysteine, as you know, is an amino acid. It's hydrophilic. There's amino acid transporters for them to be absorbed. Otherwise, they largely would not be absorbed. So we said, okay, if we're going to go down this route, let's at least hypothesize that cysteine could maybe inhibit an amino acid transporter that could be important for the drug. even though for this case, that was never really a solid idea. Nevertheless, when we start thinking about it, we're looking at the permeability values. And I should also note one of the references in the guidance, there is some overlap between at least this thinking and at least those experimental values that are in that paper. They looked at a number of different antioxidants, including cysteine, actually. And where our mind went, at least, is there are known amino acid transporters, SLC1A5 and SLC3A. These exist, including in the intestine as well as the kidney. One of them, 3A1, is actually known to be a transporter of L-cysteine. The other one is known not to be a transporter of L-cysteine, although L-cysteine can modulate it or inhibit it. But meanwhile, the drug is not known, and by inspection at least, doesn't look like an amino acid. It's not expected to be a substrate. So we didn't think, thinking in this area, we didn't think transporters would be a major risk. Okay. So overall results support that any of the 10 antioxidants up to at least 10 milligrams could be added without modulating passive drug permeability. Admittingly, looking at active transport potentially is a huge task. As you may know, there's over 400 SLC transporters and about 50 ABC transporters. Okay, and just a last topic here. Route 2 and back did some, over the summer, she did some tablet experiments involving diclofenac potassium, which is known to be a class 2 drug, although we've previously indicated that it actually represents low biopharmaceutical risk. It's a weak acid, but otherwise it's completely absorbed. I think in the next slide I show some solubility data. Incidentally, it's known to be suspect to nitrosamine formation. So here's the solubility data. Kind of looks like many other weak acid, small molecule drugs where it's, if we look at that last column, drug dose over solubility, it would take 50 liters to dissolve a 50 milligram dose. But then once you get to pH, I don't know, maybe around 5.8 or 6 or so, it becomes, gives the appearance of a highly soluble drug. And the drug is known to be very well absorbed in many different formulations involving many different excipients. So what Route 2 did is she made a couple simple formulations, so-called formulation A and formulation B. If you look on the right-hand side, formulation A involves microcrystalline cellulose as well as lactose. Formulation B involved microcrystalline cellulose and dibasic calcium phosphate dihydrate. So compared to lactose, an insoluble excipient. And then she made tablets without antioxidant as well as with AA is ascorbic acid, sodium bicarbonate, cysteine. So those are the 3 test formulations, so to speak. She made tablets. They seemed to perform just fine. They had the right dose, and they disintegrated very quickly. And the guidance doesn't mention, maybe how to do dissolution comparison, but for better or worse, we did F2 testing. And as I think you may know, there's some uncertainties about how to select values for F2. And here's a little table here, which is described in a publication that others have already observed this, right? So M9, the BCS guidance, is a little bit more restrictive in selecting data points. And the 1997 guidance has different language, leading one to choose different more data points. And in fact, even there's different ways to read the same language, actually, as you may know. So here are the dissolution profiles for so-called formulation A, and they all look pretty much the same. And you can read the table on the right-hand side. For example, the one with ascorbic acid, comparing it versus the one with no excipient at all, with no antioxidant at all. They in general were 50 or higher. I think the F2 by M9 was a little bit less than 50, but it just seems like the profiles are the same. Again, this is not a large CMC change, adding 10 milligrams of ascorbic acid, compared to maybe reading from the BCS guidance where there's potentially massive differences in formulation and processing that could be envisioned when you compare test and reference. Same sort of thing for B. Now, formulation B involved an insoluble excipient, large quantities of dicalcium phosphate, so there was cone formation, and that was consistent with the dissolution data sort of stopping around 80%. And then in the last 30 minutes, when the RPMs were increased, much more than the majority of that unreleased drug was able to dissolve. Nevertheless, when you at least calculate F2 values, there was no difference between added with an antioxidant and not having an antioxidant. Okay, and I think that's it. Thank you very much for your time.
Excellent presentation. This is what we want to hear from research, right? FDA-funded research. There is no impact on permeability. This is kind of a soothing song or sleeping song to our ears, and everybody wants to hear that. So anyway, our next speaker is Dr. Rong Wang. She suddenly got sick, got some flu-like symptoms, and severe coughing, so that's why she couldn't make it in person, but we have recording of her presentation. So we'll have her presentation now. Thank you.
Good afternoon. My name is Rong Wang. My talk today is focused on bridging pre- and post-change drug products in generic drug applications impacted by NDSRIs through case studies. The disclaimer. This presentation reflects my own view, not FDA's view or policies. In this presentation, I'm going to briefly talk about NDSRIs, common mitigation strategies, NDSRIs impacted drug products, guidance on BE data for formulation changes to mitigate nitrosamine issues, and 5 hypothetical case studies. As you have learned from yesterday and today's talks, NDSRIs are nitrosamines with structural similarity to APIs, often arising in drugs with APIs containing secondary, tertiary, or quaternary amines, especially when exposed to nitrites during manufacturing process or storage of a drug product. To address NDSRIs, applicants have several options. They can control impurities in APIs, adjust manufacturing processes, choose excipients with lower nitrite levels, add antioxidants, or modify the pH of the formulation. Often, more than one of these strategies are used together to address NDSRI. Many drugs such as antidiabetics, antihypertensives, antibiotics, and antidepressants have been found to contain NDSRIs. This slide lists some examples of commonly seen drug products that are impacted by NDSRIs. Applicants can make changes to the drug product to reduce or prevent nitrosamine impurities, and there are key guidances that outline the necessary BE data to support these changes. The first guidance covers API changes, while the second and third are the SUPAC guidance for IR and MR drug products, which address manufacturing, formulation, and other changes in the drug product. The fourth one is the FDA's recently published guidance, Control of Nitrosamine Impurities in Human Drugs. This latest guidance also introduces an alternative B approach that may be used to support specific reformulations. The new guidance recommendation for an alternative B approach is based on FDA's research. Briefly, the research about effects of antioxidants on intestinal permeability of BCS3 model drugs, and the research about effects of antioxidants in drug products on the intestinal drug transporters demonstrates that the addition of certain antioxidants does not affect absorption of BCS3 drugs. The results are also considered appropriate to extrapolate to BCS1 and BCS2 drugs. Physicochemical properties of drug substance and drug product release mechanism also play a role when considering whether an alternative B approach is appropriate to support the formulation change. Also, the alternative B approach does not apply to narrow therapeutic index drugs, orally disintegrating, or buccal dosage forms. The table on this slide summarizes the BE data recommended to support formulation changes in different scenarios. In general, when applicants reformulate by adding a pH modifier or an FDA-researched antioxidant at no more than 10 mg per dose, it is recommended to submit comparative dissolution data between the pre- and post-change products in multi-pH and QC media. This applies to immediate release solid oral dosage forms or oral suspensions of BCS 1, 2, and 3 drugs. However, when situations are beyond this, additional in vitro bridging studies or even in vivo data may be necessary. These cases will be handled in a case-by-case manner. Next, I'll present 5 hypothetical cases. The first case I'd like to discuss involves an API source change for a fixed-dose combination IR tablet. It is important to note that changing the API source is a common strategy to mitigate nitrosamine formation. However, it's typically just one of several strategies to prevent the NDSRI formation, since NDSRI are often linked to the API or its fragment. Relying solely on an API source change usually cannot entirely eliminate the risk of NDSRI formation during manufacturing or storage of a drug product. In this case, we have drug X, which comes in 3 strands that include components A and B. The original bioequivalence studies conducted on the highest strands were found to be adequate, and the formulation of 2 lower strands are proportional to the biostrands. All 3 strands show rapid dissolution profiles. However, the DMF for the original API of component A was deemed inadequate due to concerns related to NDSRI. The DMF holder did not address the nitrosamine deficiencies and opted to close the file instead. In response, the applicant proposed to switch the API source for component A. So the applicant was recommended to manufacture new batches of the drug product using the API from the new source. Some key points regarding the BE data needed for the API source change. The nitrosamine impurities in the original API of component A are the only outstanding issue in this application, with no other concerns like cGMP non-compliance or data integrity problems. Regarding the mitigation strategy, the only change being made to the drug product is the API source change. No other modifications are involved. Also, the original biobatch manufactured with the original API is considered adequate from the OPQ perspective, with the exception of the nitrosamine issue. Both components A and B are highly soluble, according to the principle of the guidance for post-approval changes to drug substance. Comparative multi-point dissolution study using QC method is needed to support the API source change when API is highly soluble. Therefore, the applicant is recommended to submit dissolution data with QC method, comparing pre- and post-change drug products for all 3 strands. In case 2, we have an approved delayed release capsule, drug Y, which comes in 2 strengths. The applicant reported that NDSRIs were detected in all tested batches, exceeding the acceptable limit. The applicant proposed reformulation as the mitigation strategy in their supplement. The applicant proposed to add alpha-tocopherol to the drug-loaded pellet and decrease the amount of sugar spares to compensate the amount of alpha-tocopherol. The applicant included pre-change and post-change comparative dissolution data using QC method for both strengths. Here's the figure of the formulation design of this delayed release drug product. As you can see that the antioxidant was added to the drug layer and the amount of sugar spares was reduced in the inner core. Some key considerations for BE data to support the change. First, per the applicant referenced literature, the drug substance of drug Y belongs to BCS2 class. The delayed release formulation consists of immediate release inner layer of drug and outside layer of enteric coating. There is a separation layer between the drug layer and enteric coating. In terms of reformulation, the applicant has proposed adding alpha-tocopherol, an antioxidant studied by the FDA. The amount used is less than 10 mg per dose for both strengths. To maintain the capsule weight, the applicant is also reducing the sugar spares by only 1% of the total weight. This change is considered as a SUPAC level 1 change. Also, alpha-tocopherol is added exclusively to the drug layer, which is not considered compromising the integrity of the enteric coating. The MDI of alpha-tocopherol does not exceed the level found in FDA-approved products for similar use, and there are no other changes in manufacturing process or API either. Based on the assessment elaborated in the previous slide, the alternative B approach recommended in the guidance can apply to this case. Since the applicant only submitted comparative dissolution data in QC media, the applicant will be asked to submit the pre- and post-change comparative dissolution studies in multi-pH media. Case 3 is a fixed-dose combination matrix-based extended-release tablet. APIs are component A and component B. The applicant reported that NDSRI was identified in API of component A and also the finished drug product. Applicant proposed to change the API source of component A, add 1.5 mg of ascorbic acid to the formulation, and change from wet granulation to dry granulation in their manufacturing process. The applicant submitted comparative dissolution data between pre- and post-change drug products in QC and multi-pH media. This figure shows the changes made to the matrix-based ER tablet, an API source change, the addition of an antioxidant, and modifications to the manufacturing process. Some key considerations for BE data to support the changes in this drug product. Ascorbic acid, an antioxidant studied by the FDA, has been added in an amount under 10 mg per dose. The MDI of this antioxidant does not exceed that of FDA-approved products. However, since this drug is a modified release dosage form, the alternative BE approach may not apply due to the complexity of the drug release mechanism. According to the SUPAC MR guidance, the change in the manufacturing process from wet granulation to dry granulation is considered a level 3 change The applicant also changed the API source for component A, but with no other issues such as CGMP non-compliance or data integrity concerns. Given the extent of these changes and the complexity of the extended release formulation, it is recommended that the applicant conduct an in vivo BE study comparing the reformulated test product with the reference standard. In case 4, drug V is an IR tablet that was approved through the BCS1 waiver approach. The applicant reported identifying nitrosamine impurities in finished lots of the drug product. To address this issue, the applicant proposed adding L-cysteine as a nitrite scavenger and replacing the high water content grade microcrystalline cellulose or MCC with a low water content grade. They also reduced the MCC amount to compensate for the addition of L-cysteine and included a desiccant in the bottle to reduce moisture. In the supplement, the applicant provided comparative dissolution data for both strains, comparing the pre- and post-change formulations in multi-pH media. In this case, the drug substance is a BCS1 class. The drug product is rapidly dissolving. The added L-cysteine is less than 10 mg per dose and is not considered affecting the absorption of the drug product. The MDI of added cysteine in both strains is below that in the FDA-approved drug product. The grade change in MCC is considered a level 2 change per SUPAC IR guidance, and the amount change of MCC is considered a level 1 change. Addition of a desiccant in the bottle is not considered affecting the quality or performance of the drug product, and no other changes were made in the manufacturing process or API. The applicant submitted multi-pH dissolution data are comparable between pre- and post-change drug products. Therefore, the post-change drug product is deemed bioequivalent to the RLD for both strains. In case 5, the applicant submitted a BCS1 waiver request for drug M. The submitted data to support BCS1 classification were deemed adequate. However, nitrosamine impurities were identified during shelf life. In response, the applicant proposed a reformulation, which included adding a pH modifier sodium carbonate in the core tablet, switching the disintegrant from cross-carbamylose to starch, and changing the MCC grade to the one with lower water content. The amount of MCC was also reduced to accommodate the addition of sodium carbonate. To support these changes, the applicant conducted a comparative dissolution study in multi-pH media for both pre- and post-change formulations. The figure in this slide shows changes made to 3 excipients in the formulation. In this case, the drug substance meets the BCS1 class criteria. The addition of sodium carbonate is not considered to affect the drug absorption. Switch MCC grade to low moisture content grade is considered a level 2 change per the principle of SUPAC IR guidance. The amount of MCC was reduced to compensate the addition of the sodium carbonate. The disintegrant makes up 1.9% of the total tablet weight, and dissolution testing can detect the impact from the change in the disintegrant. No other changes are made to the manufacturing process or API, and the MDIs of sodium carbonate and starch are within FDA-approved limits. The comparative dissolution data in multi-pH media for both pre- and post-change products are sufficient to support the formulation changes. BCS1 waiver request can be granted in this case based on the totality of the data, including solubility study on the API, comparative multi-pH dissolution data between original test product and RLD product, and comparative multi-pH dissolution data on pre- and post-change drug products, and citation of greater than 90% absolute bioavailability in the ROD labeling. In summary, many generic drug products, both approved and pending, have been affected by nitrosamine impurities. To address this, applicants can adopt multiple mitigation strategies. The guidance published this September introduces an alternative B approach for some reformulated products in nitrosamine-impacted applications based on FDA's research. Adding small amounts of specific antioxidants or pH modifiers is not expected to change the absorption of BCS 1, 2, and 3 drug substances. To use the alternative B approach in bridging the drug product changes, it is crucial to consider not only the extent of the changes, but also the physical-chemical properties of the drug substance and the formulation release mechanism. The acceptability of the alternative B approach for a given application is a fact-specific analysis conducted during scientific assessment. Alternative BE approach may be applicable to drug products that are not BCS 1, 2, or 3 IR drug products, for example, delayed release dosage form within certain contexts. And applicants are encouraged to engage with FDA through meeting requests or control correspondences as appropriate to obtain feedback on the proposed bridging strategy for scenarios not covered by the guidance. Lastly, I'd like to thank my management and colleagues in the Office of Bioequivalence for their support and the CRCG FDA Workshop Planning Committee for this opportunity. With that, I'll end my presentation. Thank you for your attention.
Another excellent presentation with very relevant case studies. So our next speaker is Dr. Hansong Chen. So he will be presenting additional case studies, but for new drug product.
Thanks, Anna. Good afternoon, everyone. I will briefly share two case studies from the NDA submissions. This is my disclaimer. So in my presentation, I will first briefly introduce principle for bridging pre-change and post-change formulations. Then I will introduce the first case study. That's from original NDA submission. Then the second case study is from NDA supplement. Finally, we'll talk about the conclusion. I believe my colleagues already talked about nitrosamine guidance recommendation for alternative approach to measure BA or demonstrated BE. So I just briefly repeat the principles. If the reformulation involves 10 milligrams of antioxidant or less, for IR drug products containing BCS class 1, 2, or 3 drug substance, multiple meeting comparative dissolution data might be considered to support formulation bridging. For the IR drug products containing BCS class 4 drug and modifier release drug products, validated IFC, PPPK modeling and simulation, or B study may be needed to support the bridging. So this is the first case study. It's from our original NDA. Drug product is drug X, IR tablet, 2 strengths, 10 milligrams and 20 milligrams. Drug substance X is highly soluble and it's either a business class 1 or 3 drug. Initially, applicant developed an analytical method, to detect nitrosamine level. Also set a specification, for nitrosamine. But those, those measures are not enough. The Siemens, our Siemens reviewers found, that those acceptance criteria are not acceptable. So applicant was requested to perform a root cause investigation to identify the source of NDSRI and take appropriate measures to reduce or prevent the formation of the NDSRI. Applicant come back saying that, they reformulated their drug product by adding ascorbic acid less than 10 milligram to the formulation. Before they resubmitted this our NDA, the applicant contacted us to explore the possibility of the bivalve for the reformulated drug product. So our feedback is, it's possible if they can provide a sufficient justification that the small amount of ascorbic acid doesn't affect the absorption of the drug X. So in the resubmission, they provide the solubility data for drug X. As shown in this table, drug X is highly soluble across the physiological pH range. This table shows the formulation comparison between the pre-change and post-change formulations for 10 milligram strength. Applicant only made 2 changes. They add 1.65 milligram ascorbic acid to the formulation. That's a 1.5% change relative to the core weight. To keep the whole tablet weight unchanged, they also reduced the amount of one phenol by 1%. So totally, the total change is 3% relative to the core weight. They also provide comparative dissolution data in multiple medium. The left figure is for 10 milligram. The right figure is for 20 milligram. As shown in these 2 figures, all batches can achieve 85% dissolution at 15 minutes. So those batches are considered similar without calculating F2 or similarity factors. During the NDA review process, applicant also submitted an IND amendment to claim drug X product is business class 1. However, the in vitro permeability data results failed to support the high permeability classification of the drug X. Specifically, the permeability data are not consistent across different tests. In one test, the data show drug X has high permeability than the high standard model drug. In another test, the data show the drug X has a lower permeability than the high standard drug. In this resubmission, applicant used literature data to justify that the addition of the ascorbic acid in the formulation has no impact on absorption of drug X. They also provide the results of 2 previous B studies. The left figure is the comparison of orally disintegrating tablet and IR tablets, the proposed drug in this NDA. even these two drug products have totally different formulations. The B study showed they are equivalent. So the right figure is the comparison of sprinkle capsules and IR tablets, the proposed drug. So again, this is drug products have two different components and the composition, but B study showed they are equivalent. So the applicant concluded, significant excipients changes in the formulation of the drug X do not affect the absorption of the drug X. So the conclusion for case study 1. So drug X is a highly soluble drug substance, and the drug product is very rapidly dissolving. Comparative dissolution testing result demonstrate the original and reformulated drug products exhibit a similar dissolution profile in multiple media. The amount of the ascorbic acid added to the formulation is less than 10 milligrams as recommended by our R2 guidance. Also, FDA research shows that it didn't affect permeability. So the rest of formulation, composition, and the manufacturing process was the same. Two BAB studies show that significant excipient change in the formulation of the drug X do not affect the absorption of the drug X. So therefore, a scientific bridge, based on this alternate B approach was demonstrated. So the second case is from NDA prior approval supplement. Drug product is drug Y delayed release capsule, 4 strengths, 20, 30, 40, and 60 milligram. Drug substance Y is a business class 2 compound. In the original supplement, the applicant proposed, controlling this nitrosamine impurity in the API and the drug products by setting specification. However, CMC reviewers found, this proposed specification are not acceptable because it exceeds acceptable intake limits. So to address the CMC deficiency, applicant reformulated the drug product to reduce the nitrosamine concentration to meet the AI criteria. Again, before they resubmitted this NDA supplement, applicant contacted us to explore the possibility of a biwaiver for the reformulated drug product. So our response is yes, it's possible. Yeah, you can use data to justify whether the addition of ascorbic acid will not affect the oral absorption of drug Y. We also specifically recommend that you can use literature or database precedents or established IVFC or your PPK modeling and simulation to support the biwaiver request. In the resubmission, applicant did propose the IVFC model. However, that model was not acceptable because it has several fundamental flaws. For example, 2 batches used for model development have exact same formulation, exact same manufacturing process, and have similar dissolution profiles. So, those 2 batches should not be used to develop IVFC according to our guidance. Applicant also provide comparative dissolution data. They provide that data was generated on 60 milligram in QC media. That's not enough. We expected the, conduct comparative test in for all strains, also in multiple media, right? For the delayed release drug product, they have to, in other condition, according to our guidance. Applicant did, provide a very nice justification that the ascorbic acid has no impact on the absorption of drug Y from a PK perspective. For the absorption, this report, the literature showed that at the low intestinal concentration of ascorbic acid, active transporter is the primary mode of absorption. In contrast, drug Y has a high permeability, and passive permeability is main absorption mode. So, the transporter effect is clinically insignificant in the intestine, liver, and brain. So no interaction, from absorption perspective. Distribution, they said ascorbic acid is only 25% protein bound and is widely distributed in all tissues of the body. Drug Y is highly protein bound, binding primarily to the albumin and the alpha-1 acid glycoprotein. Metabolism, this I have a different, this drug Y and ascorbic acid has different metabolism pathways. And the major biotransformation pathways for drug Y involve oxidation of the left cell ring, followed by conjugation and further oxidation. In contrast, ascorbic acid is metabolized hepatically, reversibly oxidized to dehydroascorbic acid, and metabolized to negative ascorbate to sulfate and oxalic acid. So no interaction between the from the metabolism perspective. So finally excretion, they said drug Y and ascorbic acid have different excretion mechanism. Ascorbic acid is excreted through the kidneys. Drug Y is cleared primarily by metabolism with minimum renal excretion. So, and they said interaction with renal transporter is unlikely. So they said, okay, no interaction, from excretion perspective. And they also justified that ascorbic acid has no impact on the in vivo performance of drug Y from a PT perspective. They reported that ascorbic acid and drug Y have different mechanisms of action. Ascorbic acid is used for dietary supplement, and the drug Y is indicated for psychiatric condition. So even they provide very nice justification, but we still, our conclusion is the scientific bridging is not established because the following deficiencies. The first, the IMFC model was not acceptable. The second, they should conduct comparative dissolution testing for all strains. Also, they should conduct additional comparative dissolution testing. For example, dissolution testing in the other pH media for buffer stage, and also to additional agitation space according to our super guidance, MR super guidance. So in the second resubmission, they repeat comparative dissolution test in QC medium for all strains. As shown in this figure, the pre-change and post-change are similar based on the calculated F2s. But still, this data are not sufficient because we request more comparative dissolution test in other conditions, right? So to our surprise, applicant conduct a B study, that actually we didn't expect to conduct a B study. They conducted this B study between the reformulated drug Y, delayed release capsule, and the listed drug. So the B study results show they are bioequivalent. However, according to our super guidance, also nitrosamine guidance, this study, does not meet our expectation. They should compare pre-change and post-change, not post-change with this drug. But finally, we still accept the B study result. We consider the following reasons. We conduct a cross-study comparison. We find both pre-change and post-change formulations have similar point estimates and the ratios of LSM for log transferred PK parameters. You can, sorry. You can see this table. Also, applicant provide sufficient data, in previous submission to justify the presence of ascorbic acid in the post-change formulation doesn't affect disposition of the drug Y. Also, applicant, said, it's not ethical, to administer pre-change drug in healthy subjects because of the nitrosamine issue. So based on the totality of the data, we think a scientific bridge has been bridged between the pre-change and the post-change formulations. So the conclusion is, for the IR drug products, comparative dissolution data in multiple medium may be sufficient to bridge reformulation. For modified release drug products and also BCS class 4 drugs, validated IFFC, PVK modeling and simulation, or a new B study may be considered to support formulation bridging. I would like to take this opportunity to thank my colleagues, Bhagwant and Oppo. They provide very valuable input during my my assignment assessment. They also provide very valuable input when I prepare my slides. Chen was the secondary for my second case. Henning is my current DD. Really, thanks for help and support. Thank you for your attention.
Another excellent presentation. Our next speaker will present virtually on nitrosamine risk assessment for BCS class 4 IR drug product and MR drug product with the use of PVK modeling. Dr. Sivacharan Kollipara.
Thank you, Alam. Thank you, Alam, for inviting me to talk in this particular workshop. My name is Sivacharan Kollipara. I am working as a team lead in the biopharmaceutics department at Global Clinical Management at Dr. Reddy's. And before I start my presentation, I would like to thank FDA as well as CRCG for giving me an opportunity to talk about the nitrosamine risk assessment for BCS class 4 molecule containing IR products as well as MR products and use of in silico prediction tools as well as the PPPK modeling. So before I start my presentation, I would like to indicate that the opinions expressed in this presentation are my personal opinions and they do not represent the statement or opinion of Dr. Reddy's Laboratories. So this is the content of the presentation. We will see the nitrosamine's overview in the context of the regulatory landscape, which most of the presenters have touched upon. And then we would like to talk about the in silico assessment of nitrosamines, which we can do using our in-house tool that we have developed, which can calculate the CPCA calculation, and it can be used to flag the nitrosamine risk assessment during the early development. And further, coupled with the in-house tool, we will talk about the opportunities of PBPK modeling that it can offer for the NDSRA risk assessment in case of BCS class 4 IR products as well as the MR products, followed by the conclusion. Coming to the nitrosamines overview in the regulatory landscape, I think the US FDA guidance that has come as latest in September 2024 has been touched by most of the presenters in this particular session as well as in the previous session. Just to recap, this particular guidance provides alternative BE approaches for BCS class 3 molecules when the mitigating agents such as antioxidants are used less than 10 milligram, and also the fact that the extrapolation also can be performed to BCS class 1 and class 2 drugs, thereby providing some alternative approaches due to the NDSRI reformulation. And this guidance also talks about alternative approaches for BCS class 4 containing IR drug products as well as for the MR drug products with the help of IVIVC as well as by using the physiologically based pharmacokinetic modeling. So the question that we are trying to address through this presentation is that can we leverage the PPPK modeling approaches to avoid the repeat bioequivalent studies for BCS class 4 containing IR drug products as well as the MR products? And the second question that we would like to answer using this presentation is that how do we utilize the in silico tools for nitrosamines risk flagging at the early during the drug development? So basically, we are trying to come up with an entire in silico workflow from the beginning of the development until the end of the development, wherein there is a possibility to avoid the repeat BE studies for BCS class 4 containing IR products as well as the MR products. So in silico assessment of nitrosamines, and in this particular part of the presentation, we would like to talk about the in silico tool that we have developed in Dr. Reddy's, which is basically leveraging the use of digital tools. So in this particular tool, a lot of things can be done. The first task that this particular tool can do is the risk assessment, wherein it can identify the risk of NDSRA formation, and later it can also calculate the CPCA, and it can also derive the acceptable intake limits based on the CPCA rule. And followed by that, this tool also can calculate the maximum allowable nitrate as well as the total nitrate content, which we can limit in the drug product so that the NDSRI formation can be reduced. And further, this tool also can act as a knowledge management, wherein we can gather the data of different types of API as well as the formulations in order to have a comprehensive nitrate database. So this is the snipping of this particular tool. And this tool, what it basically does is that it assigns the risk of the API based on the propensity or the reactivity to form the nitrosamine impurity. Again, there is an automated workflow that this particular tool uses. So for example, we have taken selumetinib as one of the example to demonstrate this case study. And the moment we type selumetinib in this particular toolbar, it automatically takes the structure from the literature, and then it calculates various aspects such as risk category as well as the CPCA prediction, and it can also give the maximum allowable nitrate limit for this particular molecule. And again, you can see from here that once the structure has been imported into this particular tool, it can identify the functional groups which are prone to an NDSRA formation. And in case of selumetinib, there is a secondary amine as well as the other nitrate containing functional groups are identified. And out of these two functional groups, secondary amine is prone to have high severity for the NDSRA formation. As a next step, what this particular tool does is that based on the CPCA rules defined by the US FDA guidance, it identifies the alpha hydrogens and it detects the activating as well as deactivating features. And then it also flags the exceptions and provide the acceptable intake limits. And this entire process happens in an automated way. Again, just to give an example of selumetinib, and as we have discussed in the previous slide, that the secondary MN group, it poses high risk for the NDSRA formation. And following the flowchart that is defined by the US FDA guidance for the calculation of acceptable intake limit, there is no tertiary MN that is present, the tertiary hydrogen that is present near to this functional group. And based on this, the tool is calculating that the severity is under the class 5 and the acceptable intake limit is 1500 nanogram per day. And further, the tool also possesses a capability to calculate the maximum allowable nitrate limit, which can be set as a limit in the drug product. So overall workflow of this particular tool is that once we have the compound that is kind of inputted into this particular tool, the tool flags high risk in case of molecules with secondary or tertiary amines. And then it follows the US FDA guidance, and it considers whether the structure is applicable as per the CPCA. And if it is not applicable or the CPCA rule is not applicable, then there is a low risk for the nitrosamine formation. And if there is a if the structure is applicable for the CPCA formation, then the tool basically goes through the literature to understand if there are any acceptable intake that is published in the EMA guidance as well as US FDA or the health authority guidance. And if the acceptable intake limit is published, then this limit is what is shown by the software. And in case the acceptable limit is not published, then the CPCA assessment based on the structure as well as rule-based model can be done, and then the acceptable intake limit can be derived. Very often it is also case that not only the API but also the degradation products can also form nitrosamine impurities. And again, the similar flow chart that we are following for the API is also followed for the degradation products in case the degradation products have potential to form the nitrosamine impurities. And again, the CPCA rule applies to which functional groups and it doesn't apply to which functional groups. It is also considered in this particular tool as per the US FDA guidance. So now we have derived using this particular tool the acceptable nitrate limit, and we also have identified the molecules which are prone to NDSRA formation, and there is a possibility of reformulation. So how do we extend this tool further to PBPK modeling to identify or to make an approach for the risk of avoidance of BE studies? So in the next session, we are going to talk about the nitrosamine risk assessment for BCS class 4 products as well as the MR products. And coming to the BCS class 4 molecules, so what we have done in this particular case study is that we wanted to come up with a PBPK modeling strategy which can be used to avoid the repeat bioequivalence studies due to NDSRA reformulation for the BCS class 4 molecules. So what we have done is that we have taken 40 BCS class 4 molecules, and then we have entered the structure of all these BCS class 4 molecules into our in-house tool. And for all of these 40 molecules, we have determined the severity, CPCA potential, as well as the acceptable intake limit. And out of this, the tool has shortlisted 15 molecules has high severity category. And out of this 15 molecules, we have selected 5 molecules which have exhibited high severity for the nitrosamine formation. And from the modeling perspective, we have selected those 5 molecules wherein transporters are involved in the absorption. Because BCS class 4 molecules are little notorious with respect to solubility, with respect to permeability, as well as the dissolution, and the involvement of transporters will make the case much more complex. So we wanted to evaluate the cases where the transporters are involved. How can we use the PPPK modeling to come up with a proper risk assessment for the BCS class 4 molecules? And the 5 identified molecules in this case are selumetinib, furosemide, hydrochlorothiazide, edoxaban, as well as bosutinib. So for all these molecules, we have developed the PBPK model, and we have included the transporters, and then we have done the detailed parametric sensitivity analysis to understand the impact of permeability as well as the transporter kinetics on the Cmax and AUC. And finally, we have tried to come up with permeability alterations and the exposure assessment and try to define a permeability safe space. So these are the set of molecules which we have utilized in the PPPK modeling, and these are the transporters involved, and this is the modeling workflow. So we started with the model development with all physicochemical properties as well as the disposition and also the transporter kinetics, and we have optimized the transporter kinetics wherever it is applicable. And then we have validated the model at least at 2 different doses, and the cases where the intravenous dose is available, we have validated the model, and the oral doses, at least 2 doses, we have validated the model, and the prediction errors of the acceptable model should be less than 15%. And finally, we have done the parametric sensitivity analysis for permeability, and then we also have altered the permeability by plus or minus 10 and 20% to understand how the permeability alterations will impact the exposures. So this is the model validation data for all the 5 molecules. And again, we have validated the model at least at 2 doses and as well as at the intravenous dose if it is available. And in all the cases, the model described the pharmacokinetic linearity very well. The prediction errors for Cmax as well as the AUC are acceptable. And that's where the models were utilized for further application part. So what we can see in this particular slide is the parametric sensitivity analysis for the first molecule adapsoban. And we have done the parametric sensitivity analysis for the Cmax as well as the AUC parameter. And this sensitivity analysis was done for 3 different parameters: effective permeability and the Vmax and Km for the absorptive transporter peak glycoprotein. In case we are using any of the excipients such as antioxidants or pH modifiers or the surfactants, sometimes they also have propensity to alter the kinetics of the transporters as described by James Polli in the previous presentation. And that's where we have done the sensitivity analysis not only with respect to permeability, but also with respect to the transporter kinetics. And in addition to this, other factors which can play a role in the bioequivalence or the impact of excipients for BCS class 4 molecules is the solubility as well as the dissolution rate. And along with the permeability as well as the transporter kinetics, we need to consider the solubility as well as the dissolution rate. So what we can see from the parametric sensitivity analysis is that permeability, of course, has a significant impact both on Cmax as well as on the AUC. The transporter kinetic parameters did not impact the Cmax much, but they have significant impact on the AUC, which is also understandable. Further, we also have evaluated how the permeability alterations can impact the T/R ratios. And at least for edoxaban, within plus or minus 20%, the permeability alterations did not impact the T/R ratios against the original formulation. The next example is for the bosutinib. And again, in case of bosutinib as well, permeability increase has led to increase in the Cmax as well as AUC. And the transporter kinetics have a little bit of impact on the AUC, but not on the Cmax. And interestingly, in case of bosutinib, permeability alterations have significant impact. And when the permeabilities are altered between 20%, there is clear bioinequivalence that has been observed. And we further assess the impact of the dissolution on bosutinib, and when we alter the dissolution by plus or minus 10% as well as 20%, we try to assess what is the impact on the bioequivalence. And interestingly, when the dissolution rate is reduced by 20%, there is a clear bioinequivalence is observed. So how does this PBPK models can integrate all these aspects is that it can consider the dissolution, it can also consider the solubility alterations, and it can also consider the permeability alterations coupled with transporter kinetic alterations, and thereby it can give a real estimate of impact of excipient on the bioequivalence part. In the next example, selumetinib, again, the permeability alterations have significant impact on the Cmax, but not on the AUC. And the transporter kinetics have significant impact on the AUC. And again, this molecule is little robust with respect to the permeability alterations. And within plus or minus 10% as well as the 20%, there is no impact on the T/R ratios that has been observed. The last molecule, furosemide, and here we have studied the impact of permeability, and clearly permeability also impacted the Cmax and AUC, and the permeability alterations within plus or minus 20% have clearly impacted the T/R ratios. So with these 4 molecules, we have seen different scenarios, wherein in few cases dissolution is impacting, in few cases the permeability alterations are impacting, in few cases they are not impacting. But through appropriate integration of solubility, dissolution, transporter kinetics, and permeability into the PBPK model, we can use this tool as an attractive opportunity to avoid the repeat BE studies for BCS class 4 containing molecules. The summary from the BCS class 4 molecules is that, as we discussed, their behavior is governed by both solubility, dissolution rate, as well as the permeability. And presence of transporter in the absorption process may possess more complexity in compared to the cases where the transporters are not present. And the impact of excipient that we are adding to mitigate the NDSRA risk has to be considered appropriately on dissolution, solubility, as well as on the permeability. And the PBPK models coupled with transporter kinetics helps us to define the permeability safe space. A representation of the safe space is what is provided here. In this case, within plus or minus 20% alterations in the permeability is not impacting the bioequivalence. So although the work that we have shown in the last couple of slides is indicating the potential of PBPK model, currently we are substantiating the model work with actual experimental data, wherein we are carrying out the in vitro CACO2 permeability studies in presence of certain surfactant as well as the antioxidants. And we are also collecting the data of the solubility alterations as well as the dissolution rate alterations because of the presence of these excipients. And we hope to we hope that this entire data set will provide us a clear-cut information on the utility of PBPK modeling for BCS class 4 molecules. The next case study that we would like to provide is for the modified released formulation, and how do we use the PBPK modeling for the modified released formulation to avoid the BE studies due to the reformulation. So in this case, the drug product is a modified released formulation containing the BCS class 3 APA at a dose level of 1000 mg. Again, this molecule is also substrate for a lot of transporters at the absorption level, and there has been a non-linearity reported for this molecule, especially in the dosage range of 500 to 1000 milligram. And the bioequivalence study was conducted on 1000 milligram, and the successful bioequivalence is achieved both in fasting and fed condition. Because of the nitrosamine formation in the MR product, there are few alterations done in the formulation, and the BE risk assessment was done using the PBPK modeling. And again, in the model, we have incorporated the Vmax Km of the transporter at the absorption level. And this is the representative dissolution data of the pivotal test and reference formulation, which are in the green color as well as in the blue color curve. The extreme dissolution profiles represents the boundaries of the dissolution, upper specification as well as the lower specification. And the dissolution profile of the reformulated drug product is what is shown in the red color curve. And the model has been developed using this particular QC media, which is also biopredictive in nature. And the model has been validated both in fasting and fed conditions. And the model validation is considered to be successful as indicated by the table. Now, although the red color curve, which is the resolution profile of the reformulated product, that seems to be near to the pivotal test and pivotal reference formulation, and even the F2 is matching. In this case, we try to evaluate how we can use the PBPK modeling. So in case of PBPK modeling, basically we have defined the dissolution safe space, and the dissolution specification that we have shown in the previous slide. They are clinical relevant dissolutions, and the actual drug product is to be taken in the fed condition. And this is the virtual bioequivalence result in the fed condition, wherein the lower dissolution specification as well as the upper dissolution specification are bioequivalent to each other. And the fact that the dissolution profile of the reformulated drug product falls within the lower and upper specification, that is also considered to be deemed to be bioequivalent. So how the PBPK modeling has helped in this case is that once the clinically relevant dissolution safe space is established, any dissolution profile of the reformulated product that is falling within these boundaries is also considered to be bioequivalent. In the present case, one point is to be noted that the excipients in the reformulated product, they may not have significant impact on the API because of the high solubility nature. But in other cases where the excipients have impact on the solubility, or if the excipients have impact on the transporter effect, they need to consider it appropriately in the model. But overall, this particular case study highlights the use of NDSRA risk assessment for MR products using the PBPK modeling and the relevance of the clinically relevant dissolution specifications. So with these two case studies, the current US FDA guidance on the nitrosamines highlighted the waiver opportunities for BCS class 3 and class 1 molecules as an alternative BE approaches. And there are also opportunities provided in terms of in vitro in vivo correlation or the PBPK models for assessing the risk of BCS class 4 IR containing drug products as well as the MR products. And we have seen through the detailed case studies that for BCS class 4 molecules, PBPK modeling coupled with solubility, dissolution rate, and the transporter kinetics, they helps us to establish the safe space for the permeability, and they can be attractive choices to justify the avoidance of repeated BE studies. And in case of MR products also, we have seen the utility of PBPK modeling coupled with the transporter kinetics. And further, as I have mentioned, the future direction of this work includes actual permeability, solubility, and dissolution impact. and integration of this particular data into the modeling in order to have a full-fledged PBPK modeling. And overall, the PBPK modeling continues to support the waiver opportunities for nitrosamine BE risk assessment, and they help to avoid the repeat BE studies, and they can enable the faster approvals. So I would like to thank Tausif Ahmad as well as Mahendra Chogle from Biopharmaceutics team, Karthik Parsa and Priyans Pandya from Digital and Process Excellence team at Dr. Reddy's. And I would like to thank Dr. Reddy's management for providing all the infrastructure and support. And this work is a collaborative work of different cross-functional teams and regulators who have contributed to the data generation as well as the feedback. Last but not the least, I would like to thank US FDA and CRCG for giving me this opportunity to present in this workshop. Thank you very much, and over to you, Alam.
Excellent presentation from Mr. Kollipara. So our next speaker is Dr. Zhang, and she has very interesting perspective on managing bioequivalence risks for nitrosamine impacted BCS class 4 drug.
Hi, good afternoon, everyone. Thank you for joining this presentation on managed bioequivalence risk for nitro-impacted drug products, specifically focused on BCX class 4 drug substance. My name is Zhang. I'm from Division of Therapeutic Performance 2, Office of Research and Standards, and on CDER and OCD. So here is the quick outline. So first, we are going to look at the current approach for BE assessment based on the BCS classification. And next, we are going to have an examination on nitrosamine impacted products across different BCS classification. And then we are going to have an in-depth look at the BCS class 4 drug products. And final is some thoughts about future directions. So I think now everyone is very familiar with the recent FDA guidance regarding the alternative BE approaches. As we know, the BE reassessment for nitrosamine reformulation poses a risk to healthy subjects. Therefore, alternative BE approach is currently our priorities. FDA advocates all the different kind of alternative BE approaches to support the reformulated products. And as indicated in the FDA guidance, the BCS classification is the key for BCS class 1 and 2 and 3. Comparative dissolution can serve as a surrogate for BE assessment. However, for BCS class 4, more advanced BE assessment tool, such as the PBPK, PBPM modeling, which is just presented previously, that can correlate the in vitro to in vivo, would be recommended. However, the guidance cannot cover all the possible scenarios, so the guidance actually opened the rooms and spaces for scientific discussions and also risk-based approaches with scientific evidence to justify the reformulations with different BCS classification. So let's look at what the specific challenges that BCS class 4 drug substance presents. So as we know, the BCS class 4 is categorized based on the low solubility and the permeability. Because of the low solubility and no permeability, it's going to be very challenging to predict in vivo performance. And traditional dissolution testing may not be very reliable to predict in vitro correlate to in vivo. So therefore, a more robust, advanced dissolution testing and with PBPK modeling can open a very useful and helpful pathway for those BCS class 4 drug products and offer the biowaiver for those products. So here shows the distribution of the nitrosamine impacted drug products across different BCS classification. On the, as shown in this slide, the the left pie chart shows the distribution of the nitrosamine impacted products across all the IR drug products, where you can see a significant portion of the IR products affected by the nitrosamine concerns. And then another pie chart shows the distribution of those IR impacted nitrosamine products across different BCS classification. And BCS class 4 make up the notable portion, which underscores the specific considerations and alternative B approach for those BCS 4 class drug products. And let's look at the food effect and fraction absorbed for those products containing BCS class 4. And we have collected together data from the wide source of information data to estimate those BCS classification. And we examined the food effects and the fraction absorbed for different BCS classification. So based on these figures, we can see the BCS class 4 actually has variable food effects and can be positively or negatively or no effects. And for in general, for the drugs with the positive food effects usually has a higher fraction absorbed, whereas the negative food effects usually will have a much lower fraction absorbed. So with that foundation, with that data analysis, we look at the BCS subcategories, of course, within this BCS classification, and we analyze the associated risk factors and then the limitations. So this table categorizes the BCS class 4 into different classes. It's mainly in solubility limited or primarily permeability limited or dual limitation. For solubility limitation, which is similar to BCS class 2, the risk factors include the pH dependence, dissolution, and sometimes potential precipitation. And for the permeability limited, which is similar to BCS class 3, and the issues arises in general from the excipients that may affect the permeability, and also as shown before, transporters, and sometimes the absorption window could also affect the in vivo performance. So with these risk factors and also the subcategories, let's move on the findings that we found from the BCS class 3 model drugs that can be extended to BCS class 4 drug products. So the table presents the data for the BCS class 3 model drugs with the estimated permeability and the food effects, AUC and Cmax, and with different dosage forms containing same API, but all the products contain same API with the BCS class 3, mainly is BCS class 3 drug substance. And the research on those BCS class 3 findings can be extended to BCS class 4. So for those BCS class 4 drug substance where they have a similar permeability, then the findings that antioxidant would not expect it to affect those BCS class 4 drug substance. And of course, if it's higher, especially the fraction absorbed is higher than those BCS class 3 model drugs, or the permeability is not a limited factor, we don't expect any or significant effect on in vivo performance bioavailability. So this is something that we can have some bioweaver argument for the BCS class 4 drug products based on the findings from BCS class 3. And so based on those factors and the findings from BCS class 3, we propose a 4-tier risk-based framework for the products containing BCS class 4 drug substance. So we start the solubility testing to evaluate whether the BCS 4 products is solubility limited. And usually the solubility is very low or positive food effects and with a higher fraction absorbed would indicate this is a pH, it's a solubility limited. And for permeability limited BCS class 4 products, we mainly compare the fraction absorbed with the BCS class 3 model drugs. If it's higher, then that means the permeability is not the limitation, and it can extend the BCS class 3 findings to BCS 4. And let's move to the tier 3 when there is a solubility or permeability limitation. And then dissolution testing is also the key. Then we need to conduct the dissolution and the different pH conditions and also simulated physiological median to determine whether there is a solubility limited factor or permeability limited factor. And if those 3 tiers indicate high risk, then also the prediction from the absorption also indicate high risk, such as PBPK, PBPM modeling, then the in vivo PK study would need it. So in conclusion here, I want to highlight the main key points is that the BCS classification is the key factor to support the alternative B approach, and the BCS class 3 findings can be extended to BCS class 4 drug products. And for dual BCS class 4 drug products, and future research and discussion ongoing, and then the directions will be how we are going to analyze and evaluate complex formulations and interactions between excipients, and how we formalize the BCS classification in order to support alternative B approaches. So with that, I want to acknowledge my supervisor and my director, Rob. He provided lots of insights for this short presentation. Also, Gang and Dongmei for their help and with the data analysis. That's my presentation. Thank you for your attention.
Excellent presentation. Our last speaker before the break is Dr. Fang. She will be presenting use of PPK modeling to assess the risks of BCS class 4 drug as well.
Good afternoon, everyone. Today I'm going to talk about the PBPK modeling for BCS class 4 drugs and case examples. Here is my disclaimer. As you all know, the guidance control for nitrosamine impurities in human drugs issued in September 2024 indicated that approaches to inhibit nitrosamine drug substance-related impurities formation include the addition of small amount of antioxidant or pH modifiers. To understand the addition of these antioxidants, the impact on the permeability of BCS class 3 model drugs, the FDA funded a series of research. Based on one of the research, we found that the antioxidant tested here, including alpha-tocopherol, ascorbic acid, cysteine, propyl gallate at concentrations tested had little or no impact on the permeability of the 4 BCS class 3 model drugs, including acyclovir, atenolol, cimetidine, and ranitidine. This suggests that reformulating drug products to include this antioxidant may be a feasible approach for reducing the formation of NDSRIs. As indicated in the guidance, these research results conducted on BCS3 drugs appropriately extrapolate to BCS1 and 2 drugs that have high permeability. However, due to the complexity of the low permeability and low solubility for the BCS class 4 drugs, this is complicated, and so validated the in vitro, in vivo correlation, PBPK or PBBM, and or in vivo BE studies appropriate to demonstrate BA or BE and support formulation change. Today, I would like to present to you some our previous experiences on using PPPK modeling to evaluate the impact of excipients, including antioxidant, on BE of some BCS class 4 drugs. This example is acyclovir. I actually presented this case at other occasions as well. This one, acyclovir, is BCS class 3 drugs, considered as BCS class 3 drugs at a low dose, but it is a BCS class 4 drug at high dose. We built the PPPK modeling for risk assessment. First, we use the IV administration and those PK data after IV administration, and we used to develop PPPK model for this acyclovir after IV administration. Then we use the PK data collected from the literature, develop PPPK model for acyclovir IR tablets, and then we validate this model using PK data and the BE studies for acyclovir IR tablet, the high dose, and to validate this model. After validation of the model, we conduct a sensitivity analysis to evaluate the effect of apparent permeability on the PK/BE parameters, including the Cmax ratio or AUC ratio between test and ROD. And then we conduct virtual BE trials to assess the potential impact of the excipient-mediated permeability change on the BE of acyclovir IR tablets. This similar as you see in Siva's previous presentation, we conducted a sensitivity analysis on the apparent permeability change on the impact of the PK/BE parameter ratio, the Cmax ratio or AUC ratio. You can see with the increase of the apparent permeability change, the PK parameter ratio is increased. And it's, we can try to find acceptable level at between the 0.8 to 1.25%. And also we conducted the virtual BE simulation. Based on the single virtual BE simulation, we found that up to 50% increase of apparent permeability, it is resulting in the bioequivalence. However, when we increase the 60% apparent permeability, there is this failed BE observed by PPPK prediction. So in summary, this case example indicated that the VBE results suggested that more than 60% change apparent permeability value for the test product due to presence of certain excipient that could, including the antioxidant, may result in the failed BE of acyclovir 800 IR tablet and the fasted condition. For other BCS class 4 drug products using PBPK and the VBE simulations, you can also detect the boundary of acceptable permeability changes for risk assessment. The second case example I would like to share with you is using PK model to evaluate the impact of solubility and permeability on PK for the IR tablet of ticagrelor. So ticagrelor is a platelet inhibitor. It is also a BCS class 4 drug. So similarly, we built the ticagrelor PPPK modeling and using the IV PK data obtained from the literature to estimate disposition parameters, and we've developed the PPPK model for IV administration of the ticagrelor. And then we input the particle size of the drug substance and the solubility, and using the Johnson dissolution model to inform the dissolution. And we developed the oral PPPK model using these data. And then validated the PPPK model using PK data from the in vivo clinical studies with different formulation, like the solution or generic ticagrelor IR tablets with different API particle size. And then we conduct a sensitivity analysis to evaluate the impact of solubility and the permeability on PK parameters. And then for this PPPK model, we conduct VBE trials to assess the risk of non-BE by using the upper limit of the particle size. This we consider as a boundary proposed by the generic drug firm. And actually, that model was used to setting the clinical relevant PSD specification. This sensitivity analysis I want to show you is to see how the permeability and the solubility interplay with each other. It can impact the sensitivity of the AUC and the Cmax sensitivity to the particle size changes. And so you can see from the y-axis indicated the AUC. The x-axis indicated the API particle radius. And from the top to the bottom, the permeability from the low to high. And then from the left to the right, the solubility from the low to high. And the different colors indicate different precipitation time. So if you look at the top panel, it is the low permeability, and you can see the AUC profile seems more sensitive to the particle size changes. You can see the profile is steeper if you compare it to the bottom panel. It means that AUC is more sensitive towards the particle size changes. And vice versa, if you look at the permeability, if you look at the solubility from the left panel, the 0.0011, that is low solubility compared to the right panel, 0.11, the high solubility. You can see the profile tends to be more sensitive to the particle size changes. The profile is more steep compared to the right panel. And if you look at the 0.011, the left panel, and from the top to the bottom, you can see AUC tends to be more sensitive to the permeability change compared to high solubility. Similar trend can be observed for the Cmax as well. So for this case example, these sensitivity analysis results suggested that various parameters, including solubility, permeability, and also the API precipitation time, can influence the sensitivity of oral absorption towards API particle size distribution. Actually, these can show us that when the permeability is low, actually it is the AUC and Cmax is the sensitivity seems more sensitive towards the dissolution as well. And so lower permeability results in more sensitivity of PK parameters towards solubility or particle size changes. Also, lower solubility results in more sensitivity towards permeability or particle size changes, as I just now described. So, taken together as a practice advice, FDA is open to alternative approaches like PBPK modeling to support demonstration of BE for reformulated BCS class 4 drug products. And conduct a sensitivity analysis using this modeling, you can determine the boundary of acceptable permeability change. This also, Siva also presented in his examples. And by incorporating the dissolution information, and this can be used for supporting your risk assessment for reformulation of BCS class 4 drug. If you have questions or if you would like to use this model-based BE alternative approaches, MIE, the Model Integrated Evidence Industry Meeting Pilot Program, can be used to seek FDA feedbacks. This concludes my presentation. I would acknowledge all of these people listed here for their support. Thank you so much for your attention.
Excellent presentation by Dr. Fang Wu. So it is time to have our panel discussion again. Is it working? Okay. We'll start our panel discussion very soon. If you can, please take your seat. And I like to introduce our 3 additional panelists. Dr. Liang Zhao. Dr. Zhao is a professor and director for the Center of Regulatory Science with the School of Pharmacy at the University of California, San Francisco. Before joining to UCSF, he served as the director for Division of Quantitative Methods and Modeling in the Office of Research and Standards. He received 2023 Gary Neal Prize for Innovation in Drug Development from ASCPT in recognition to his contribution to clinical pharmacology and pharmacometrics. Our next additional panelist is Dr. Bhagwant Rege. Dr. Rege is the Division Director for Pharmaceutics in CDER OPQ at FDA. His division at FDA is responsible for assessment of clinically relevant in vitro release specifications for drug products, in vitro in vivo correlations, or IVIVC, physiologically based biopharmaceutics model, PBBM model, scientific bridging strategies, biowaiver strategies, BCS classification requests, etc. Our next additional panelist is Dr. Sumit Madan. Dr. Madan is the Vice President of R&D at Sun Pharmaceuticals Industries Limited and heads the Formulation Research and Development Department for oral formulation. In his current role, he leads a talented group of scientists engaged in developing complex generic, value-added generics, and specialty products for global market. Do we have every panelist here? Okay, yeah. Yeah. Is Wang, Dr. Wang online?
Yes.
Okay, great. Okay, so first we will start with the in-person attendees question. We'll give the priority to the in-person attendees question. If you have any question, we can start with that. And then we'll entertain some virtual question, and if time allows, then we'll have additional discussion.
Hello. This is a question regarding the antioxidants where you discussed you can add the antioxidants up to the maximum daily exposure. And that is based, I assume, on the levels that are reported in the IIG. So, for example, there's quite a bit of discrepancy depending on the dosage form in the IIG. Tocopherol, for example, has a range of 1 milligram as a maximum daily exposure to 246 milligrams, depending on whether it's a tablet or a film-coated tablet. Can you please clarify that the MDE that is listed in the IIG is the maximum daily exposures? Are those set limits? Are you looking at those more carefully to see if those can be expanded further?
Hi, my other hat has to do with the IID. So I'm trying to get out of that business. But actually, the maximum daily exposures that you see in the IID, that is the maximum amount that has been used in products. But when you talk about the research that was done, I believe the maximum that was used was 10 milligrams per unit dose, correct? So if you're trying to stay within the limits of FDA's research, no matter what it says in the IID, 10 milligrams would be within our research. If you're going to go outside the research, then other types of BE studies might be needed. There was something else I was going to tell you. It went right out of my head. Go ahead. Ask your other one.
That gives clarification. So is the FDA looking anything beyond 10 milligrams for those studies? Because in some cases, 10 milligrams might be insufficient to fully mitigate nitrosamine formation per dose, that is.
I believe the guidance specifies if you're going to go about 10 milligrams, then you may need to provide evidence depending on the BCS class of compound of that additional level that you're using, that it doesn't affect permeability, and some additional justification will be needed in that case.
So 10, but 10 seems to be a bit arbitrary. Have you looked at for further higher levels is the real question here.
So basically, why we use this 10 milligram, there's this reason for that, just kind of an assumption basis on that that's 2% of antioxidants in the assumed unit tablets or capsule, that's 500 milligram. And because, only 2%, 1 to 2% of antioxidants, that will be really significantly inhibits the nitrosamine formation. So that's the reason why we use this 10 milligram. Seems that's a magic number, but actually, and if you in the guidance we already mentioned in the category number 2, if you use an antioxidant that is more than 10 milligram, and as Bhagwant already mentioned, that is you need to most of the cases for BCS 1, 2, and 3, dissolution may be already sufficient, but for this, BCS 1, 2, that is already sufficient. And but for the BCS 3, that's a supportive information should be needed if you are beyond that scope. Yep.
So may I ask a relevant question? So in our guidance, we mentioned that if it is more than 10 milligram, for BCS class 3 drug product, we may want to conduct or to provide the permeability or transporter activity data, right? So how about the additional novel approach like PVK modeling or IVIVC? Do you think that that is another possibility for BCS class 3 drug, IR or MR? I know that MR, we already mentioned that PVK modeling or IVIVC is possible, but how about for IR drug product, if you can clarify that?
So you are asking if they're using more than 10 milligram?
Yeah, more than 10 milligram for BCS class 3 IR drug product.
Yeah, it would be useful, but even as data input into our PBPK model, you'll still need to show what effect on permeability the extra amount of antioxidant will have, either passive permeability or transporter activity. So that will need to be included in the model. So. Okay, yeah. And also, I believe that even for developing PBK model, you need some sort of permeability data to feed the model, right? So, okay.
Sorry. The nitrosamine guidance that has the formulation updates and then an alternate BE approach is all for oral solid dosages. Is the intention for the agency to come out with recommendations for other dosages, specifically like injections where we can't necessarily modify the pH because then we wouldn't be matched to the RLD?
Okay. So yeah, as you all know, because it's a nitrosamine impurity impact the oral drug products a lot more than other dosage forms. And so that's the reason why we made really great effort and try to have this develop this recommendations for this oral solid oral dosage form and plus this oral suspension. For some other drug products and other dosage forms, across the board, there are relatively limited number gets impacted. And if you want to communicate, in general, FDA, you will evaluate this reformulation case by case. Definitely, you are encouraged to contact FDA and for the further discussion.
This is Rong. So I just want to add a couple of points. So you mentioned the injectable. Actually, for parenteral product, especially for solution, there is, it's a waiver approach for for establishing BE for this type of product. So the alternative BE approach may not be applicable here. And also for injectable, there is a regulatory requirement of Q1, Q2 sameness of the formulation. So if applicants opt to add, for example, antioxidants to the formulation, then I think it's a separate issue. So whether this is a approval, waivable product or not, and as I said, it's a separate issue and needs to be further discussed. But usually, for Q1, Q2 sameness is the strict regulatory requirement for injectables. Thank you.
So I was going to say that one of the nice things I saw in the guidance is that you call the nitrite scavengers as antioxidants, which it's true, and these are exception excipients. So one good thing would be that formulating injectables, which are Q1, Q2, antioxidants are considered like preservative antioxidants. These are exception excipients. So hopefully and many people kind of assume that by calling these antioxidants, we are supporting a lot of the reformulation. But I think the previous speaker was talking about the pH, because a lot of times you see that the pH of an injectable is very important, and it's in the label of the innovator, and you have to follow. So, trying to modify that can cause issues, and I think she was probably indicating that will the agency consider those kind of variations?
Thank you for the comment.
Yeah. I had another question. I saw that a lot of you, again, it's an excipient thing, but Susan, kind of IIT, but not exactly. We do see that even FDA used caffeic acid and ferulic acid, and as well, they were my favorites. I have used them a lot in my past life, but they are very benign material. And we are just naming them. There are plenty of other antioxidants which are not in the IIT, but in small amounts may not be risky. And I mean, one appeal that I will be, making, and I think most of the industry will, that maybe to accept reformulation with these rather than real non-clinical testing, but paper documents which could show the safety at that level, would be great.
Now I remember what I was going to tell that last gentleman. Yeah, the FDA undertook some research to test some antioxidants without actually looking, I think, at what was in the IID. In fact, some of those would be considered novel excipients, and they wouldn't be appropriate for use in a generic drug formulation. And if they were to be used in a new drug formulation, they would still have to provide safety information on those novel excipients.
That is exactly where I was trying to come, that is considering the depth and the vast expanse of this issue, and some of these being very, very potent, is there a possibility that, internally you could talk to see? There is phenomenal amount of safety data on some of these antioxidants for the amount they are going to be used, that instead of asking for a battery of non-clinical testing, killing dogs, cats, and other animals, could we submit a paper document to justify the limit, even in a new drug?
So this is a farm tox question. There is a guidance on novel excipients, and that guidance does have some flexibility in it. If there's actually data, that can be submitted. But I suspect for one of these excipients that was used in FDA studies, but it's not listed in the IID, there's no proof that it's been used in an approved product, the first applicant that wants to use it has to provide something to the agency to show that the safety of it is supported, and for that route of administration and that context of use.
If I could add some, okay, I think these two considerations, IIG limit and the level of antioxidant in the reformulation are addressing different questions. IIG limit is set to conform or to verify, to prove that that level of inactive ingredient used in the formulation is generally considered as safe. The level of, the antioxidant added in the reformulation is to say that, yes, I'm adding this level of antioxidant in the reformulation, and it does not change the permeability absorption of the drug. But we put it together because the number-wise, we want the lowest to ensure both considerations are addressed.
Yeah, go ahead. Sorry. Go ahead.
And with regarding to other dosage forms, I think this morning you raised up a topical product, a nasal drug product. The majority of, or a part of, at least a part of inhalation drug product, nasal drug product, topical product, are locally acting drug products. So we pretty much rely on a weight of evidence approach, considering, in vitro studies, formulation similarities, as well as PK studies, but PK study is not considered as the direct evidence to support equivalence, but as an indirect and a downstream bioequivalence approach. So from that point of view, that the alternative approach may not applicable to those locally acting drugs when it formulated as inhalation or topical drug products. Thank you.
Very clear. The reason I said IID is because there are numerous antioxidants which are not in IID but would be phenomenal nitrite scavengers, and that's why I was asking if there is a tricky, easy way that we could push it in with paper document. That was my question. I do understand that, the path of least resistance is to choose one from IID. Thanks a lot, Bing. That was a very good explanation for the other formulations.
My question for Sivacharan, is there any plan to maybe publish is not the right word, but share that software that you guys have built so that the rest of the industry can leverage it? And I guess secondly, to our FDA contacts here, is there something that you could consider that has these rules built in so that it makes it easy for the end user, and also from a documentation standpoint to say, I've put the structure in, I've got the CPCA value, and it's kind of, this is your tool and it's told me what it is, so that justification from our end becomes a little bit easier?
Yeah, thank you for that question. So in terms of making the software available for the public, so basically we have built this software based on FDA guidance August 2023. All the rules and regulations that we have put up in this software is entirely based on that particular guidance, with a little bit of artificial intelligence in terms of getting the data from the literature with respect to acceptable intake limits and CPCA values going through various databases. Putting the software in the public domain, that call needs to be taken care after getting the regulatory, internal intellectual property department approvals. At this point of time, I don't have the question, but I think this software, again, just to summarize, the software is based on the US FDA August 2023 guidance on the NDS array acceptable limits. Each and every calculation and other aspects are appropriately considered in this particular software. But just to summarize, my answer, I need to check internally for making this software publicly available. Maybe for the other part of the question, I would request any FDA representative to answer that.
So since you asked this question, so I want to inform something to our audience, that we have a pathway that is model master file. So any applicant or any CRO or any interested party can submit their own model with some validation, and we will make it publicly available that this software is available, and other applicants or other companies, if they want to use that, they can reach out to the model master file holders, how they can collaborate or can use it. So that is another pathway that can, Dr. Reddy's lab or other applicants can think of. Dr. Liang Zhao, I want to ask you one question related to model master file. So since you were in FDA for a long time, probably more than 10 years, and you are the main architect for the model master file. And we have discussed these topics in different public workshop, how model master file can be used in the landscape of generic drug and new drug application. But this is nitrosamine, it's very specific. So do you see any opportunity here for the model master file?
Yeah, thank you, Alan. Thank you for posing that question to me. Also thank the whoever asked the question. I think it's today I'm wearing different hat, so I can be a little bit liberal.
I believe that is a good thing, because you are outside of FDA and you can give your more perspective what outside of FDA people can think of, right? Yeah.
So I think a model is not only a model, it's more like a summary of all the knowledge. You integrate every knowledge together for regulatory use, like how to integrate the interplay between permeability, dissolution, solubility, all the things. They play together. You observe a very interesting pattern, as Dr. Fang presented. Only at the low solubility part, you see sensitivity towards particle size, for example. All those patterns cannot be easily recognized by just using the conventional thought. Therefore, model is really the ultimate tool to summarize all the knowledge to support even more important regulatory use. For example, the earlier question, is the acceptance limit set like a 10 milligram? Can we expand that? I will say, so if we integrate every knowledge, I really appreciate the colleagues in FDA in the past have achieved so transparent. Every decision science-driven. So I feel so proud to be a part of FDA. So I will say, anyone in the outside of FDA can propose, here is the model. So for example, FDA published a paper for NDMA that even you take, from food or take a huge amount. So I think we did an internal modeling effort. Then certainly there's a human clearance, right? Then there's also endogenous generation of those substances. So we eat kimchi, right? Sometimes. So can we integrate all the information to convince the agency here could be another more practical new limit? I really think FDA is very open-minded, welcome those ideas. Also for model master, say Siva developed a very sophisticated PBPK model. How can that be shared? Then I think FDA devised a new mechanism on top of the model integrated evidence meeting mechanism. You can submit a model master file. The owner there are multiple benefits with the model master file that is shareable. Then another applicant, another sponsor can utilize the same approach. For FDA, then such a big organization, then reviewer A have reviewed and accepted part of the model for certain regulatory use. Then reviewer B can easily get the history of the modeling use and then enjoy enhanced consistency, efficiency, productivity. So I think the exact purpose of model master file to make model generally shareable across the industry applicants without much duplication in effort, without much duplication in the regulatory agency, then we gain efficiency from both sides. You can certainly publish the model, make it open source, but the problem with that is that you don't know whether already gained regulatory acceptance or not, what the regulatory opinion on that. So every communication with FDA associated with cost, time, and effort, right? You need to develop a package, you need to wait for FDA to schedule a meeting for you. But if people can have model master file, I think that people can all enjoy the latest advance in science and the regulatory acceptance. I think that we need to pay attention to that mechanism. Many of us here, the audience, well, promoting that mechanism. Hope we can all pay attention to that.
Yeah, excellent point. I think that we have seen a lot of interest on using PBK modeling for particularly BCS class 4 drug product, how to manage the risks, because our guidance was very clear, is very clear about class 1, 2, 3 drug, but there are some risks are not very clear maybe about the BCS class 4 drug, but here is the opportunity of PBK modeling or IVIVC correlation, how we can use this. So my question to Dr. Fang, since you presented your PBK modeling, that what are the considerations we should take account when we develop the PBK modeling and how we can assess the risks, if you can briefly describe your opinion.
Yeah, thanks for the question. During my presentation, actually, I also presented the modeling. If we want to build the model, we need the in vitro testing, including the permeability test and also the in vitro dissolution. You see the interaction between the solubility, particle size, and also permeability for the BCS class 4 drug products. So you need to consider of testing the permeability. For example, if you want to reformulation for the BCS class 4 drugs, for that antioxidant, if it's already being studied by FDA, yeah, you may use that result, but if it's not, you may need to consider conducting the permeability testing and put it into the model and build it and validate the model. Ideally, you need to have the in vitro data and also the in vivo PK data to validate your model. If not, you may consider using the theoretical non-BE batch or then put into the model to test the discriminating capability of your model, then use the sensitivity analysis and to find the boundary of the permeability change or the dissolution safe space, and also do the VBE simulations and for supporting your risk assessment for reformulated BCS class.
Excellent point, yeah.
Oh, thanks. Amazing amount of work. I mean, this is really incredible to see how much FDA is doing to support this. But unfortunately, I got to drag us back to a little bit more operational issues. One question, a little bit easy, more clarification. The 10 milligram value, I think on the slides today it said per dose. Is that maximum daily dose, unit dose? Could make a difference.
Okay, so this information really comes from the research project FDA sponsored, and so our foundation or the rationale to design this study is just based on the 10 milligram per unit. But in terms of for your specific drug products, you need to consider and whether, for this MDD, for the daily dose, maximum daily dose, and in terms of the antioxidant amounts added into each unit, whether that is really beyond this maximum daily exposure. So definitely there are multiple factors you need to consider that. That should be lower than, totally that should be lower than the maximum daily exposure.
Yeah, when we talk about 10 milligrams, it's pretty much, I think I would interpret it as a single highest dose. So at any given time, we are taking a tablet, and that's your single highest dose at that time. You should not exceed 10 milligram in that tablet. If you're taking 2 tablets at the same time, then you should not exceed 10 milligram combined in 2 tablets.
Yeah, okay.
Yeah.
All right, I understand. I have another question, which is even more operational, and if this is beyond your scope, let me know. But you've done a lot of work, industry is doing a lot of work to address this. I would expect a lot of change requests. Have the agency invested? Are you aware of any changes in operations to address that flood?
So when we revised the guidance and we said that you could send in, what did we say, CB30 for specification changes, we were considering how many of these requests we were going to get. When we wrote the part of the guidance that addressed formulation changes, and we said, you know, what can we do to make it faster, easier for applicants to submit this data to us, that's when we started looking at what we can do for BE studies. Can we make these faster and easier? And that's when we undertook the research in order to support that sort of downgrading approach to make it faster and easier. So yes, we were really worried about being flooded with supplements. Yes.
Okay, thank you.
So thank you for the great discussion. So we also received a couple online questions from virtual audience. So the first question, my guess, is for Dr. Susan Zuk. So this is a clarification question. So Dr. Susan, you said like pay attention to the August 25 date as all submission for changes relating to the reformulation for nitrosamine issue. So the question is, will the agency not accept PAS, post-approval supplement for reformulation after this date?
Well, we did set that date. I would say if a company can't, if a company has an intention to submit something and they don't think they can submit the data by that date, they certainly can contact the FDA and explain their intention to submit the information. We kind of drew a line in the sand that August 1st, 2025 date seemed far away when the rail guidance was published, and we thought, well, this is, you know, doable. We still think it's doable, but if manufacturers are starting to have difficulty with it, certainly they can contact us and let us know what their intention is as far as reformulation.
So next question is also for Dr. Zuk, and also maybe Dr. Lu. So the question is, regarding reformulation, if minor moderate changes are made to formulation or manufacturing process to control nitrosamine, so is one batch with data sufficient for filing with a commitment to generate additional data in future batches? That is sufficient?
I don't know. I'm not quite sure, and maybe Dongmei understands better, but I think it's about the stability studies. Is it about stability
It's
studies?
not specific here, maybe for some testing, or I remember.
Want to give it a try?
Okay, so in terms of the stability data in my presentation, actually in the guidance, we recommended 3 batches. Why is that? Because, you know, if you have this as the SUPAC guidance open, then you will notice that if, you know, the changes and for the changes, actually, if there is a significant body of the data not available, then we will request 3 batch of the data for the stability. Because, you know, NDSRI right now really fall into this kind of scenario. in-house, and we have some data now, but just compare it back several years ago, almost nothing. And also in terms of each individual company, and they may have their own mitigation strategies, and we don't know whether that individual strategy is really effective or not, because that is really for the different cases and for the different formulation design and for the different drug products. So based on this, and we have developed this recommendation. So 3 batches, in terms of the stability. Yeah, but if you have further questions, I think definitely we also encourage industry to contact FDA.
Thank you, Dongmei. So the next question is for Dr. James Polli. So why is ascorbic acid studied, but not sodium ascorbate, given that ascorbic acid is acidic and may provide a pH that is favorable for nitrosation?
Yeah, this is a chemistry question, is that right?
Yeah.
About ascorbic acid or sodium ascorbate.
Yeah, why
facilitating some of the reactions that we've been talking about the last couple of days. Yeah, I think in general, right, these reactions require acidic conditions. I suppose one could worry about, adding an acidifying agent to a formulation, how that could actually, not mitigate at all the problem, at least in the solid state. But I haven't done any research on that. I don't know if anyone else has.
I agree. It's kind of contradictory, right? You are using ascorbic acid as antioxidant, but then it gives acidic microenvironment. So you may have to use sodium ascorbate in some cases, but I mean, it depends on, in terms of stability, mitigation of NDSRI for one molecule may not require the same approach as what is required for another molecule. So you may have to do some pre-formulation work to figure out which is the best antioxidant to use.
Thank you both. So the next question is for Dr. Hansong Chen. So why adding antioxidant? Is there any need to adjust other excipients to maintain the same core weight of the tablet?
Sorry.
So one reformulation with addition of antioxidant. So is that necessary to adjust other excipients to keep the consistent weight of the tablet core?
At least from my, no, we didn't request them to keep the whole tablet unchanged. It's an applicant decision. That's an applicant decision.
Yeah, that is correct. We don't mandate that. It's really the applicant's formulation decision. All we request is the addition of addition of any antioxidant. That should be the only change that they are making to the formulation in order to use the alternative BE approach. If they are making any additional extensive changes, then the regular CPAP provisions might apply.
Thank you. So next question is for Dr. Kollipara. So the question is, in case of tertiary or quaternary amines, does the tool evaluate the different potential NDSRI depend on the product of nitrosative cleavage?
Yes, the tool can predict and evaluate all the potential and multiple NDSRI based on the nitrosative cleavage. It can be different degrees of the functional groups that are present, the amines that are present, but the tool can give details of all the NDSRI impurities. And as I mentioned previously, it is a replica of this tool is in background, it is working based on the principles of August 2023 FDA guidance, so it can give not only for the drug product but also for the potential impurities.
Thank you.
I wanted to go back to the 8-1 date. Maybe I got confused. Could you clarify exactly what the expectation is by the 8-1 date? Is it that the change request must be submitted to the FDA? Is it must be approved? Or is it that the product going into the market must meet the regulations? I mean, what exactly is the intention or the thinking behind that 8-1 date? What must be done by that date exactly?
Okay, so I think literally the guidance says that steps 2 and 3 for the NDSRI should be done by 8/1/2025. And so that would be you finish doing your confirmatory testing, and you should be, if you're going to submit something to the agency, it should come by then. I don't think anybody's going to be looking to see, did it come exactly on this day? But basically, we're telling industry that confirmatory testing should be done, and you should be, making your plan to perform some sort of mitigation, whether it's reformulation, changing your source of excipient, whatever you have determined the root cause to be. I hope that's clear.
Somewhat, but I guess to follow up, in the event that we're waiting for the approval to come through, are we allowed to continue shipping the product, or are we in that situation of we have to wait for the approval to come through before we can
So you're
ship?
talking about a product that has not been approved yet, and...
No, it's approved, but it's a change request, right?
Okay,
So
so
do we-
you're waiting for the supplement.
Right.
Wow, I think that needs to be addressed with the agency, whether you can ship something or not. I mean, you're talking about a product that's already approved to be on the market, right?
Right.
So I think you really should address it with the division, that you're dealing with for the approval.
And then slightly follow up, in your chart where you had, you do confirmatory testing and then you're in the 3 zones of less than 10, between 10 and 100 and above 100 or something like that. The last block was you need to do a change request or some type of submission. Could you describe what the scenario you think it would be for OTC monographs where there is no submission required?
Right. So with OTC monograph products, of course, there's no approval, right? But we still expect them to do this kind of work. So we would expect also by August 1st, 2025, that they would have addressed through confirmatory testing and making any changes that are necessary based on the root cause, any NDSRIs that are forming in their products. So it's all the same, except they don't send something to the agency and they don't need approval before they can be marketing the product.
So the other question is for Dr. Fang Wu. If VBE simulation is used to demonstrate the addition of antioxidant does not impact the demonstration of BE for BCS class 4 drug, does sponsor need to conduct type 1 error rate simulation?
So the type 1 error simulations, actually, it is to give you an idea under what simulation conditions you will control the type 1 error. So that one, we are also conducting the research for what is the best practice on the VBE simulations, and what is the how to get the estimation of subject number, what is the number of VBE simulations will control the type 1 error. So we are conducting the research for giving the the recommendation on the best practice of VBE simulations. So after that, that will be, you are welcome to submit your type 1 control and for, using simulations. And yeah, then we will we can use the MIE pilot meeting to give you the feedback. Thank you.
Thank you, Dr. Fang.
Yeah, I want to add here something. Yeah, so thank you so much for mentioning about MIE pilot program, because I think that these are so complex question that can be addressed through MIE pilot program or some other communication pathway like pre-ANDA or other communication. Control correspondence, maybe that is another pathway. But here I want to mention that this product, right, it was bioequivalence before. Now they're changing their formulation, and we have a kind of somewhat confidence on the product that was approved before. So I believe that the type 1 error or virtual bioequivalence, the stringent criteria we are thinking, maybe we need to think about more that how we can actually develop our framework for this kind of drug product, nitrosamine impacted drug product, to assess the bioequivalence through the virtual bioequivalence. Maybe the stringent criteria for type 1 error or other thing may not be applicable, or maybe somewhat we have some room to explore there.
Thank you. So we also received a couple of questions regarding the drug product quality. So the first question is, with regards to nitrites in excipients, as a lot of excipients suppliers are not willing to include a stringent nitrite limits in their specification. So would it be sufficient if nitrite is tested by the drug product manufacturer and include an internal specification or similar?
Yeah, in a nutshell, yes. It's the drug product manufacturer that has that vested interest to know what the nitrite level is, right? So we would expect that the drug product manufacturer would be the one saying, this is what we need for our product, and they should have a specification. There's a lot of information out there about various products, excipients. The LASA database has information on levels in various excipients. I mean, I think it depends on the supplier, but I recently saw it was a presentation by BASF where they were pulling data from that LASA database on the various nitrite levels and creating a similar model for formulation to avoid nitrosamines. So I hope I answered the question sufficiently.
Yeah, definitely. Thank you. So the other quality question is, if the NDSRI increases during shelf life at 36 months, so then do we still need to include the NDSRI in the specification? My question is, the question is asking about, they observed like NDSRI is like above the level, but it was observed like in 36 months.
So if I understand correctly, and I hope I heard everything you said, a trend is noticed. But I'm assuming it's still less than 10%. Is that the assumption that we have here? It's less than 10% of the AI, but there's an upward trend. Is that what? I didn't get into that detail. I would say if you see an upward trend, it would be prudent to have a specification because you don't know what's going to happen, right? But I don't, like, how high are we talking?
Yeah, how about the case, is over like an AI limit?
If it's over, then it's over. That product shouldn't be sold. It's, exceeding the acceptable intake limit. So it depends on the shelf life too, right?
Right.
Is it a 36 months shelf life or is it 24? If it's 24 months shelf life and it's within, then it's within. But certainly if you're seeing an upward trend like that, then some mitigation should be done so that the product doesn't go out of specification. Do you want to add to that? No.
Yeah. Thank you, Dr. Zhu. This question is asking like, did FDA observed discoloration or browning of the tablet when adding antioxidants to the formulation? So any advice to avoid this from happening?
So since we are not the manufacturers, we have not observed, but ascorbic acid is known to react and form discoloration upon shelf life. So it's really up to the manufacturers to look at that, see if that's the best antioxidants, or use some additional strategies like tablet coating maybe to see whether that can be mitigated or not.
I want to quickly check if we have any in-person question as well, because we are entertaining virtual question for a long time, but we are running out of time as well because we have just 1 hour and we are all tired. I know that you are listening all the day, so want to check with you that if you have any question, then we can take them. Otherwise, we can continue with the virtual question or other discussion we can have. Okay, you can go ahead, yeah.
So next question, asking, do we need BE studies if we change from white granulation to dry granulation to reduce the NDRS amount for BCS class 1 solid oral dosage form? Yeah, I'm not sure if...
Are you talking about BCS class 1?
Yeah, BCS class 1, asking.
Again, it's the types of studies. I mean, wet granulation to dry granulation tends to be a level 3 change. But if it's a BCS class 1 drug and FDA has granted BCS class 1 designation to that compound, and if the dosage form is the same, if the dose is the same, and if it's an orally administered product, you can submit data to get ICH M9 based biowaiver. So that would be sort of a different mechanism. But in general, if you're making extensive changes beyond simple addition of nitrite scavenger, you probably would need a BE study. But again, if it's a BCS1 compound, you can go through that route, M9 biowaiver route. But it does need to have formal BCS class 1 designation.
So next question, asking about if the pre-change product generates data above 10% of AI and a simple change in excipient source of the same technical grade, which resolves the issue. So, and AI data for below the 10%. So is that okay to submit all the data in an annual report?
If I understand correctly, the inquirer described a change where before the change, the level of the nitrosamine was below 10% of the AI. After the change, it was still below 10% of the AI?
Before change is above 10%.
Yeah, so if it's below 10% of the AI, it's not necessary to establish a specification limit. And as I mentioned, every time a manufacturing change is made, it's prudent to retest and make sure that these levels are staying below 10% of the AI. But it's not necessary to establish a specification as long as the manufacturer confirms that the level is below 10% of the AI.
Thank you.
So, Dr. Zhang, I want to ask you one question. Since you have presented the risk assessment for BCS class 4 drug product, so my question is that how confidently we determine whether a BCS class 4 drug is primarily solubility limited or permeability limited, and what implication does this have on the bioequivalence assessment?
For this question, to determine if the BCS class 4 drug substance is solubility limited or permeability limited, I think we can check and start with some serious evaluation, for example, solubility testing and the different pH conditions and with different medium. So if the solubility can be improved under a certain pH environment or some certain with a specific excipients would indicate is solubility limited. And also if the food effect indicator is positive, that also can indicate there's a solubility limited factor there. So if for permeability limitation, like I show in my presentation, you can compare with the BCS class 3 model drugs to see the fraction absorbed. Usually if it's permeability limited, the fraction absorbed should be kind of low or comparable to the BCS 3 model drugs. And also you can conduct the permeability testing if the permeability is still low with the improved solubility, that means indicating it's permeability limited. So how we are managing those solubility limited or permeability limited BCS4 is kind of we are taking a risk-based approach. And I think for solubility limited, the BE assessment can focus on dissolution. So using the adequate dissolution conditions and create a robust dissolution testing to see whether the formulation changes will affect the dissolution that will also affect the in vivo dissolution and in vivo performance. And permeability, and I think we can always compare with the BCS class 3 model drugs to see whether we can manage the risk based on the understanding from the BCS class 3 model drugs. And also as other presenters, they share the modeling could be a very helpful tool to predict the permeability impact on absorption. Thank you.
Thank you. We don't have a lot of time, but I want to ask one or two questions. So can you extend the alternative bioequivalence approach for BCS class 4 drug or MR drug product if we know that addition of antioxidant is not going to alter the GI permeability or absorption? Is it possible? Maybe you can, please go ahead and answer that.
So we have to understand why the recommendations were made to include immediate release products of BCS class 1, 2, and 3 drugs eligible for alternate B approach, and why we said for BCS class 4 drugs, IR products, and MR or ER products, we need additional information. So for absorption, after you take the drug inside your body, you have dissolution and permeability. So these two steps have to take place, right? So the question is, addition of these extra excipients, how are they affecting these two processes? So for that, we evaluated effect of antioxidants on permeability and showed that at least those 4 antioxidants at up to 10 milligram will not affect permeability. Then the next question is, how does it affect dissolution? And for dissolution, we are actually really thinking about in vivo dissolution. So the question is, does in vitro dissolution, is it representative of in vivo dissolution? So for BCS class 1 and 3 compounds, for the IR products, you can say with reasonable confidence that in vitro dissolution will represent in vivo dissolution. But for BCS class 2 compounds, that we cannot say it with that confidence, but the way to mitigate that risk is we know that they have high permeability, and because they have high permeability, there won't be any transporter issues. So if you shows dissolution similarity at different pHs, physiologic pHs, based on reasonable risk, we can include BCS class 2 compounds in that framework. Now, extrapolating this to class 4 and extended release product becomes complicated. So it's easy to say, oh, we are just adding a little bit of antioxidant, there is no dissolution change, so we should be allowed to use it. But these in vitro dissolution for BCS class 4 drugs and ER products may not be reflective of what happens in vivo. And that's why we have advocated use of modeling and simulation. And my personal pitch for anybody who's trying to develop extended release product is try to at least attempt to develop an IVIVC as part of your development program. Even if it is upfront investment, but I think it helps a lot when you're trying to make these post-approval changes. Even if you're not able to achieve a quantitative correlation, just an in vivo, in vitro relationship, it just provides a solid justification to make these changes.
Yeah, one last question before I wrap up this panel discussion. Maybe this question is also to you, actually. So you mentioned about IVIVC, and we don't have very good confidence that whether MR product or BCS class 4 drug product will translate the in vitro dissolution to in vivo dissolution, right? So IVIVC can be a good substitute or alternative approach. But what is our expectation? What you want to see in the development of IVIVC? Do you want the applicant to conduct some kind of in vivo study, or they can use some literature reported rich data if there is any? So any expectation, can you shed some light on that?
I mean, in general, any additional justification that is grounded in science is, we are willing to take a look at it. And as Hansong's case studies have demonstrated, we have looked at additional information from even different formulations. But as I said, when you're trying to apply modeling and simulation approaches, being reactive is a little, if you are just trying to leverage existing data and try to build or validate the model, sometimes it becomes limiting. I'm not saying it's not successful, but always, sometimes it can be successful, but a proactive approach is always easier. I mean, you have better chance of success there.
Yeah, and always remember that we have a MIE pilot program. You can submit this kind of modeling simulation question through the MIE pilot program. So I want to thank every panelist to participate in this session. We had a very nice discussion. One question? Sorry, we can take one. Sorry.
Thanks. Sorry, I wasn't sure what the scope of this question would be in terms of like what panel this would be best for, because it's kind of combining both days. But there's been a lot of discussion of like in silico modeling and a lot of discussion of the cost of some of the like in vivo, in vitro studies for determining the AIs for like novel nitrosamines, which there seem to be like more and more each day. Is there potential for incorporating some of these in silico models that are being developed for, say, like the bioavailability or like the dissolution and all that other stuff for these drug products to maybe be like extrapolated to maybe like the nitrosamines formed from them? Is there like a place for basically in silico data to help determine AIs, because these nitrosamines keep like coming out pretty quickly?
Yeah, Dr. Fang, yeah, Dr. Zhao, you want to answer as well? Yeah, contribute.
Probably, can you follow me? Yeah. So I really like that question. You know, I think when we talk about a model, I think the audience may have the first impression it's not a practical, it's not like a 10 milligram, big or less than 10 milligrams, it's not straightforward. But for BCS class 4 cases, I think doing modeling could be cost saving than actually conducting a B study. Also, the other benefit is that once you do the modeling practice once, then you could have gained enough experience with applying those skills. It could be extrapolated easily to relatively easily to other products down in the pipeline. So it's kind of worthwhile investment. Also, I think probably I will encourage FDA colleagues who use model, rather than case by case. Can we develop more like a decision tree type of a thing? Make the process more clear cut for industry to follow. Maybe there are some areas we never can figure out the physiological parameters. Figuring out the physiological parameters kind of itself is Nobel Prize, a great level of research. But can we use model to assess the risk. So we can make a conservative approach at the absence of some key modeling parameters. We assume a very conservative number that make can safeguard human health. Then we can still convince the regulatory agency to accept that approach. I think I would call to FDA colleagues to develop a decision tree to how to use model. What are the safe areas? What are the unsafe areas? To be more concrete on that.
Yeah, thank you for the question, and also I echo Dr. Liang Zhao's comment. For the in silico modeling, actually you can first you build the model. Maybe first you didn't use that for BCS class 4 reformulation. You did it for the particle size specification setting, as I presented the case, Tecagrello case. So, but you use it as a model master file, the common disposition model, and then you can use that for supporting the other regulatory purpose as well, like for evaluating the reformulation BCS class 4, or for evaluating the food impact, the permeability change impact, or your the absorption or bioavailability or BE. Those can be considered as a model master file by one applicant. But another way for using that is you can use the similar methodology or platform. If one applicant developed that model for BCS class 4 reformulation, then the other applicants can also use that methodology, similar methodology, to do the risk assessment for your BCS class 4 reformulation. Thank you.
Thank you. Yeah, very good point. Yeah, I will not take any further time. We are already late, so that concludes our panel discussion session. And I like to welcome Dr. Bingley to give a closing summary of the panel discussion.
Okay, first I'd like to ask you to join me to give a round of applause to our excellent speakers, panelists, and you audience for such a wonderful session. I'll see if I can take a few minutes to summarize the key point covered in session 3. I did jot a lot of notes in here. Okay, I will try to keep in a high level, okay? So we heard 7 presentations during this session, from which we learned the following takeaways, okay? So what we heard is that we heard the FDA updated guidance introduces a flexible approach for the AI limit. We actually stay away, well, transitions from the 29.5 old paradigm to a new flexible paradigm for AI limit, especially on the multiple source of nitrosamine impurity sources, and the LOD level of limit. And we also heard from Sue's presentation a nice flow chart for the implementation of the control strategies and also the timelines. And the FDA updated guidance also introduced reformulation as a mitigation strategy for reformulated product. And the scientific basis for the alternative bioequivalence approach is based on the earlier FDA's research, which suggests that the addition of small amount of antioxidant or pH modifiers may not significantly impact the absorption of certain drug substances. Meanwhile, it inhibits the nitrosamine formation. And based on this, together with the permeability and the solubility properties of drug substances, as well as the mechanism of drug product release mechanism, and we proposed a streamlined bioequivalence approach. Where in this approach, certain in vitro methods such as dissolution comparison can serve as a bridging for a reformulated product, and in lieu of the traditional in vivo BE approach. And from Dr. Jim Polli's study, his study on the antioxidant effect on the BCS3 drug permeability, the study indicated that the antioxidant studied show no noticeable effect on modulating the drug permeability, which actually confirmed and strengthened the scientific basis for the alternative BE approach. And then through the case studies the speaker presented on the NDA and the ANDA cases, we learned that the acceptability of this streamlined BE approach also requires scientific considerations of specific drug products. And then the speakers from Dr. Reddy shared an automated risk-based tool that can be applied in drug product development. And they also shared cases using PKPD modeling to assess various parameters, permeability, solubility, and dissolution on the drug absorption, and also the cases of MR products. And with regard to the modified release and the BCS4 drug product, we heard a lot of challenges of expanding the alternative B approach in this space. We also heard that we also explored PBPK modeling as a potential tools to support bioequivalence for these, complex formulations. And we learned that a deeper understanding of the permeability and the solubility properties of BCS4 substances and the factors that influence these properties will help to explore the applications of the alternative BE approach for BCS4 drug product. And I heard the term, safe space. I think this is an area that we can explore in the future, maybe in our next CRCG. Alum, you have a job on your shoulder. Then we run into an very interactive panel discussion. We addressed a lot of technical detail questions regarding the CMC part and also the bioequivalence part. I sort of extracted two top key takeaways from these discussions, okay? So I heard that in general, there is a desire to share the knowledge, for example, the software that Dr. Reddy developed, and also use model master file to support the model integrated bioequivalence approach. I also heard that there are some discussions on the timelines for the application submission. I heard that the industry can contact FDA when considering extension of the timeline and also the details of the timelines. And FDA looks forward and looking for changes and making efforts on means to make the application approval faster and easier. Okay, so that's actually, those are the few key points I captured from our session. And lastly, once again, I'd like to thank our speakers for your very informative presentations, and our panelists and you, our audience, for such an interactive panel discussions. And I also want to give a special thanks to our CRCG and also the organizing committees for putting up this wonderful workshop. And with that, I conclude our session 3. Now I'd like to invite Dr. David Keire, the Office Director for Office of Pharmaceutical Quality and Research, to deliver the workshop closing remarks. David, please.
Thank you for that. I know I'm standing between you and Beltway traffic, so I don't want to take more of your time than I need to. I do have some slides, so apologies for that. But I guess before I want to echo the congratulations to the organizing committee. I mean, really a fantastic scientific program. I think it's a really great snapshot of the current knowledge in this critical area, right, that's impacting so many drugs and our everyday lives. So begin with the end in mind, and this is kind of the end in mind, from my point of view, right? All our friends take these drugs. I don't know why the slides are going forward. Am I doing something wrong? I don't know, they've got a mind of their own. Okay, so, and this is really the end in mind, that I think we all have, all our friends and colleagues and people we know take these drugs every day. And we really want to get to a place where we don't have the nitrosamines there, or at least we minimize them to a safe degree. So now, we have an issue because nitrosamines are everywhere. That's established. They're in foods and cosmetics and the water, and that's because they're made from common building blocks and conditions that they experience conditions in the world that cause nitrosamines to form. So they are ubiquitous, and for NDSRIs, a lot of APIs have this issue, and they're in the world as well. But that doesn't make them okay to be in drugs, okay? Because people have a choice about what food they eat, what cosmetics they use, right? But they sometimes don't have a choice about what drugs they take. So it's an important distinction between those, bless you, between those other categories that we feel centrally at the agency. So there's a lot of impacts, of course, drug shortages, withdrawals, many information requests. We had a lot of questions today about that category, and of course delays in drug approval, which we want to eliminate. But on the bright side, right, we've learned a lot, and of course you saw some of those learnings in this session. A lot of the factors, right, and this is this kind of famous Health Canada figure that shows what we've been talking about, right? There can be process-related issues, there's supply chain issues, stability issues that are leading to nitrosamines in the drug supply, but there are, there's the nitrosamine lens available to look at these things now, right? We have a lot of new information. I just show these two, not because I participated in them, because I think they're just examples of regulators working together to share information, just like industry is sharing information on this topic. And when it gets out into the public sphere, that's where we can all use it to solve this critical problem, right? So I hope that collaboration and the work, I know it's going to continue because it's such an issue. And there's plenty of sources. There's lots of information. And we're sharing knowledge, and this information is an example of that, sharing of that knowledge. So the end in mind, right? We have things we can do. We can change our processes. We can look at the fluid bed drying step and the conditions and the air that's around us. We can look at making excipient changes and monitor our supply chain. I don't know why it's doing this. And we can make changes to formulation. All these things we've touched upon, and now that we'll turn that red thing in the middle to a green thing in the middle, all right? That's where we want to get. So I'll finish with that thought. This is the end in mind. Let's work all together to get there. So thank you very much. And thanks again to the organizing committee and to the staff and everybody that attended. Appreciate your attendance and engagement.